gone and how thick the deposit is as it's been redistributed. And we want to look now at some of the longer term recovery processes on the benthos. Why we want to do this is we ultimately want to verify and improve plume models, right? So that we can predict these things before they happen to a better degree. We also want to inform management thresholds. How much turbidity is too much turbidity before we start, potentially making too many organisms die, right? And ultimately determining response and recovery rates. We want to know what the time frame is for some of the ecosystem function to return. All right, so the initial baseline work, we were out on the Maersk launcher late 20 and early 21, so coming right out of COVID. It's an interesting time. Collected multi-cores right here and basically subsampled these at 1 centimeter intervals as a primary approach in 3 primary areas: the test mining fields, TM, sites wide of mining, and a preservation zone. This ultimately is the area that the collector will eventually mine. And this is the area that we are now using as our control. For the field collections and the collector tests, right? So this was late 2022, right before the test as a third baseline, right after the test within a month, and then a year later, 2023. So looking at, timestamps after the impact. So these black lines are those 80 kilometers of track. All the blue dots and numbers are all of our core sites. You can see there's kind of a gradient moving out this way. This is, a few sites within the track, zone 1, 2, 3, 4, and 5, generally going to the north and east, farther away from the tracks in the area that the predominant sediment plume was expected to go. And then we also have these areas in the SWM area that essentially serve as our control sites. So we've run a bunch of different analyses. We start off with just a visual description. Helps us understand where that active geochemical layer is, looking at redox changes, bioturbation depths, those types of things. I will give you a primer on how we use thorium-234, but we measured it using gamma spectrometer here in a minute. And then phytopigments, Bella will give you probably a lot of details about the methods there, so I won't get into the nitty-gritty there, but essentially using chlorophyll, phaeophytin as a fresh and degraded food source, essentially, and sediment processing proxy. And then we also measure sedimentology, grain size, X-ray fluorescence for elemental composition, and mineralogy using XRD, which you can't see down there at the bottom. X-ray diffraction. So starting off with the visual description, as I said, these areas are very understudied. So the only comparable study that we had to have any understanding of whether our collections were like anything that had been previously collected was Zawadzki et al. In 2020, where they essentially combined these top 2 horizons into what they call the active geochemical layer. So we're looking at, visually, what are the differences between these different lithologies. And we basically found that, just to orient you, most of the plots that I'll be showing, this is going to be 2022, so post-collector. This is going to be 2023, 1 year after the collector test, okay? This is the control, generally with longitude, west to east. These 2 are going to be the tracks, track year later, and then we're moving out zone 2, 3, 4, and 5 away from the track, okay? So ultimately with our visual, this is, very beginning of all of this before we do any of the chemical analyses. The visual descriptions are our active geochemical layer is limited at the top 10 centimeters in the control. Shallower at both of the track sites, which is consistent with the surface of those cores being removed, right? So we have some scouring going on there. And then we have a deeper geochemical layer, 15 up to about 12 centimeters, and it's shoaling away from the tracks, which is just what we'd expect if there was increased deposition closer to the tracks, decreased deposition as you get farther and farther away. So it's pretty consistent with what we'd expect. What's really interesting is a year later, there's still evidence of scouring. That geochemical layer is still only limited to the top 5 centimeters. And we still see, not to the same degree, increased deposition and then shoaling away from those tracks. Okay, so jumping into some of the actual data here. Bit of background on our approach to the radiochemistry here. We use thorium-234 as a particle tracer. It absorbs to organic matter, clays, ultimately they're settling to the seafloor. And so those particles ultimately scavenge thorium from the water column. Scavenging is caused by a whole bunch of different processes, which I don't have enough time to get into today. So we're going to get to the assumptions and the hypothesis here. The Clarion-Clipperton Zone has a really, really low sedimentation rate, right? It's out in the middle of the Pacific. And so the amount of particulate matter coming into that area is pretty low. And so thorium also has a very low half-life, right? It's short half-life, 24 days. So the assumption here is that the sediments out there in the CCZ should have a very low inventory of thorium activity, right? There's not much particulate matter coming in, and it's going to be gone within 24 days, 100 days of measurement, right? And so that's our assumption, right? And we are ultimately also assuming, and there's a little bit of data out there on water column thorium, that the water column has slightly higher inventories than the sediment. So if we are out there with a collector, right, creating a plume, will those particles that are resuspended scavenge what little thorium is in the water column and then ultimately provide sort of a target on where that sediment is then deposited? Does that make sense? Okay, cool. So this is what we found. This is our baseline measurement. This is a compilation of all of the sites in the baseline. The average is about 1 DPM per gram, disintegration per minute per gram. The sort of dark green color is all of the sites that were taken directly after the collector test. And we found that the sites in zone 2 and zone 3, this is 0.2 and 0.5 kilometers away, so 200 meters, 500 meters away from the tracks, there was a significantly higher amount of thorium activity. So, and beyond that, we don't see anything above background. So this, our interpretation of this is that that plume traveled about a half a kilometer away and no farther, right? That's the interpretation here. 1 year later, there's a few that are still relatively high, but ultimately not significantly higher than baseline, and definitely not quite as high as the directly post collector. And so our interpretation there is that it's slowly but surely returning to background. This was an anomalous event for this area. Looking at it spatially, tracks, those that are close, those that are farther away, and our controls way off to the left. So this is pre-collector. Post-collector, you can see that first half a kilometer away, north and east of the tracks, are the hot spots. And then post-collector, going back down to relatively close to background. All right, so that's one line of evidence. Trying to run through these as quickly as I can, sorry. With elemental composition, this is XRF data. Some of this is calibrated with ICP-MS as well. Same orientation, right, as those visual descriptions, 22, 23. We run pretty much everything from magnesium to uranium, but we have to boil this down for the managers. So we run a full analysis on matrix elements, redox elements, and metals. These ones are, this suite right here are the ones that we're presenting here. Same orientation, control, track, and then moving away from the plume. Ultimately, the control site shows us pretty much what we'd expect, a gradual gradient, right? Kind of a natural gradient of metals decreasing with depth. What's really interesting is when you look at the track site, the entire track profile looks like the downcore section of the control. Again, consistent with scouring. So you basically lopped off the top of the track site. All of that material had to go somewhere, right? So if you guys look closely, I know these are pretty small up here, I apologize. At zone 2 and zone 3, these surface units are pretty much homogeneous, right? And so this is also consistent with deposition at the centimeter scale. The top 1 to 2 centimeters are the same material at those two zones, right? And we don't see that at zones farther away. So this is consistent with all of this scoured material being deposited at zones 2 and zones 3, 0.2 and 0.5 kilometers away. So we're going to interpret that as deposition in those areas. 1 year later, we still see basically a straight line at the track site. So bioturbation probably has not recurred here. We don't have an active geochemical layer initiating again even a year later. And what's really interesting is the only depositional series that we can resolve, right, is limited to zone 2. We see actually a gradient at zone 3. We don't see that homogeneous surface unit in zone 3 like we did directly after the collector test. So the plume deposition essentially limited a year later to about 0.2 kilometers from the tracks. We're also using elemental ratios. And you don't have to be a sedimentary geochemist to notice the differential between these track sites, right? This is background, essentially, surface of those versus all of the other cores. So this again shows that there's something significant occurred at those sites as compared to all of those. Our interpretation is that this is consistent with scouring. Those values are consistent with down core portions of all the other cores. All right, looking at XRD, this is mineralogy, closely tied to elemental composition. Unfortunately, they did not collect cores for us prior to the collector test for mineralogy. So we're making a few assumptions here. Number one is that the control site is representative of what those track sites looked like prior to the control or prior to the collector test, right? So it's a representative sample of what would have been there prior to the collector test. And we're also assuming that the material that moved essentially from the top of that the track sites, we could ultimately measure at the surface of the plume sites, right? So if we see a greater admixture of what's at the surface of the control at those impacted sites, we can also add another layer of evidence that that's evidence of deposition, right? So do you guys follow that line of evidence? Okay, cool. Same orientation here. The first thing that jumped out at us is that the entire column at the track site was very similar mineralogically as the down core sections of all of the other cores. Again, consistent with just removing the top portion of that core. So scour with XRD is also consistent. And then we also see a greater admixture in the top 2 centimeters of that site located closest to the tracks, 200 meters away. That is very similar to that of the surface of the control. So we do see a greater admixture of the surface of the control mineralogy at zone 2. Again, pretty consistent with our interpretation of deposition there. What's a little bit frustrating is a year later, we still see evidence of scour at the track, but we don't see any evidence of that layer remaining at zone 2, right? So diagenetic processes, settling, dewatering, we're not sure, right? If that plume initially was a centimeter thick and ultimately has now been condensed down to a millimeter, we may not be able to resolve it. That's one possibility. So we need to sample at higher resolution is what I'm getting at. Another line of evidence that we are just now getting to, this is thesis work from Rachel Flickinger, has found something very similar. So the track, this is looking at biogenic silica. This is an area where radiolarian tests are the predominant, sediment coming from the surface. And so looking at biogenic silica is a nice tracer for that. So the track looks very similar to the down core section of the control, very similar to the other mineralogical cores and the elemental cores. She also has found in the top 2 to 3 centimeters a higher percentage of biogenic silica, so redistribution of that surface unit from the tracks at the plume sites, right? Particularly the one closest to the tracks. So right in line with what we found with the other proxies. Again, this is Bella Ionata's work. I'm not going to step on your toes too much. 2 quick takeaways. Lots of variability in baseline. Baseline is really, really important. So take a baseline sample right before whenever the stressor is going to happen. That's really important to your interpretation. Number 2, we found a higher concentration of chlorophyll A at those sites that are less than half a kilometer away. So independently, all of these were done independently before we put them all together. This also supports our interpretation that there was a depositional event within a half a kilometer from the tracks. Pardon me. All right, Pam, here are the forams. I'll go quickly through this. This is primarily Brian O'Malley's work. He's taken the lead on this, but it's a great pairing with all of our geochemical measurements. So early publications from this area found that about a third, right, 30% or more of all of the epipelagic or epibenthic fauna, were foraminifera. And so we're like, well, we've got some background in forams. Let's see what we can do. So we have now analyzed 77 cores, nearly 400,000 individuals, over 1,000 different morphospecies, and this is now the largest quantitative foram survey in the area. So we're pretty proud of this. What's great about it is we're trying to focus on useful things for the ISA and the managers. So we've identified first order opportunists, recolonizers, and second order recolonizers to try and figure out what the succession would be, right, based on previous impacts, oil spills, ash falls, that type of thing, that we would expect to see after this type of impact, right? So that way we can hopefully help them monitor and manage this a little more clearly. So trying to put all of this data into one slide, these 4 slides come from a report that's 240 pages. So I tried to condense it way down. This is the track, zone 1, 2, 3, 4, and 5, moving outward. 5 different measures of benthic health, essentially, using foraminiferal assemblages. The reds are significant negative impacts. The blues are significant positive impacts. Green and yellow are somewhat neutral. And essentially, you guys can see just coloration-wise, most impacted area in all 5 of these proxies was the track, right? No surprise there. But then you also see a gradation going from reds and oranges out to blues and greens within about 2 to 3 kilometers, right? So this falls right in line with where we see the geochemical impacts, right? So the biology is responding to those geochemical changes. Another cool spin-off was that we found a lot of forams that were not in our typical biofaunal fraction, right? We found a lot of macrofaunal forams and also 250 different species of monothalamus foraminifera. So these get as big as megafaunal, right? 6 to 10 centimeters, and they provide their own little host environments for other organisms. So really cool, you know, new species and that kind of thing. Several that are undescribed, so we're excited about that. All right. Getting to the takeaway message here. So for those of you that fell asleep, you can wake back up again. So we really are kind of arguing here that this multi-proxy approach is essential in this setting and many other sort of event or impact settings. We've learned a lot of nuances from each individual line of evidence, and we've tried to do them independently before we start to interpret them as a whole. By chance, they came together and pretty much agreed with our, you know, a whole holistic interpretation. But we found that there was scouring, right, on the centimeter scale that was visible even a year later. And we found that the spatial extent of that collector test plume was limited to about 0.5 kilometer. Previous models had it ranging hundreds of kilometers in some cases. So we have a pretty good, a much better idea of how to scale these types of collector test plumes. And we see that it's limited, at least as far as our geochemical evidence is concerned, within about 0.2 kilometers 1 year later. So we are currently working with several folks, DHI is a major one, to verify some plume models and improve their models. And then we're also providing this for the ISA to provide thresholds for the amount of turbidity and the amount of sediment plume that can be allowed before we see major deleterious impacts to benthic fauna. And then now, as we're getting years away from this collector test, we can start to understand the long-term impact and response and recovery rate. All right, with that, I want to thank all those folks that funded us, folks that helped us in the lab and in the field. And I want to thank you all for bearing with me as I rushed through all of this. You have time for questions, or we keep moving? One question? Okay. Got time for one question. I'm happy to chat with you all afterwards too.
Sorry.
Yeah.
For the benthic species, is it all done visually, like taxonomer goes there or they do eDNA?
As of right now, this is all visual. Yeah, so we are now at the point where we're starting to do molecular characterization of them as well. So when we, especially for the new species, that will be part of the species description. Yeah, the DNA level. Yeah. Did that answer your?
Yeah.
Okay, cool. All right.
Just a comment for the students. Our lab is host for the International Marine Mineral Society. I'm its president-elect. Laura is its executive director. And we're also holding the next underwater minerals conference out at St. Pete Beach, November 9th, 2025 at the Serrano Beach Resort. We're most happy to have student volunteers to help put that on. So if you're all interested in this, contact me and Laura, and we're working with Patrick on some of these projects as well.
All right, thank you all. Appreciate it.
All right, for the sake of time, I think we're going to skip our first morning break and go right into the first set of student talks. So I apologize for that. All right, so our another room. Yeah.
The other rooms are basically vacant now. Excuse me, where can grab the chairs?
You can see if the other closets are open. I'm not sure.
Oh, okay.
Other chairs here. You're gonna do. All right, everybody, we can get settled in. We're gonna get started on the student oral presentations. And so our first speaker today is Lydia Ruggles, who is a master's student with Dr. Maggie Brisbane, and she's going to be talking to us today about B vitamin requirements and phycosphere interactions of a hab-forming dinoflagellate. So with that, I'll hand it to Lydia.
Hi, everyone. Morning, everyone. I'm Lydia Ruggles, and I'll be discussing my research, which I've been working on with Maggie this past almost 2 years now, on identifying the B vitamin requirements and phycosphere interactions with the harmful algal bloom dinoflagellate Pyridinium bahamense var. So Pyridinium forms large blooms and produces powerful neurotoxins, also known as saxitoxins, in estuarine waters globally. In Florida, the blooms started occurring more recently, as blooms started occurring after 2002. Blooms normally form in the Indian River Lagoon and a little closer to home, Old Tampa Bay. These estuaries are similar as they both have variable season or temperatures and salinities, long residence times, and are proximity to human development. But what controls pyridinium cell abundance in these estuaries? Temperature, salinity, and nutrient loading are often correlated with many harmful algal blooms. However, it is not annually correlated with pyridinium cell abundance in Old Tampa Bay and the Indian River Lagoon. We do know that micronutrients can limit phytoplankton growth, similar to many macronutrients. Many harmful algal bloom dinoflagellates require at least one B vitamin for growth. We know that bacteria can synthesize B12 and supplement phytoplankton B12 to support growth. And many harmful algal bloom dinoflagellates require phycosphere bacteria to supplement B12 to support their growth. Therefore, I hypothesized that vitamin B12 availability and bacterial interactions influence pyridinium bloom formation in Florida estuaries. To test this hypothesis, I have 3 objectives. My first is to determine if pyridinium requires B12. I will then determine if pyridinium recruits specific microbial communities. And then I'll determine if these microbial communities can supplement B12 to support pyridinium bloom formation. So to test my first objective, 2 strains of pyridinium were collected, one from the Indian River Lagoon and one from Old Tampa Bay. These were established in the lab to create pure cultures of each isolate. The Old Tampa Bay isolate was inoculated into 2 vitamin treatments, one missing B12 and the other one containing B12, which I will refer to as replete, and that is our control. This was done in triplicates, and every 48 hours I measured chlorophyll A, as chlorophyll A fluorescence is a good proxy for biomass. At exponential growth, I serial transferred these cultures into the according media, except for the one missing B12, as those were failing to grow. This was a big surprise for us, as the pyridinium without B12 showed no growth at all. And if we look at the x-axis, we have our time of every time a chlorophyll A measurement was taken. On the y-axis, we have chlorophyll A fluorescence. And red is our media treatments when it's missing B12, and the yellow is our replete treatments. So why is it not growing when it's missing B12? So then I was like, I wanted to determine how much B12 it required. So I created different 10% serial dilutions of the highest concentration of B12 to create bi-media formulations. I then measured chlorophyll A every 48 hours. We found that it needed greater than 3.69 picomolars of B12 to grow. If we compare this to other saxitoxin-producing dinoflagellates, it is higher. And then if we compare it to the Florida red tide Carenia brevis, it is substantially higher. So again, this pyridinium shouldn't be bacteria-free, so there should be, theoretically, bacteria supplying B12 for their growth. So I wanted to test the initial presence of bacteria in my Indian River Lagoon strain and my Old Tampa Bay strain. To do this, I used 2 test broths to test the presence of bacteria, and our Old Tampa Bay strain tested negative in both bacterial tests. However, Indian River Lagoon tested positive in both of them. And we also have our controls to make sure our methods were working. I then filtered all of the triplicates from serial transfer 2 on particle-associated and free bacteria size filter paper. I did the same thing with my Indian River Lagoon isolate. I used these filters to extract RNA, and I targeted the V3 and V4 region of the 16S rRNA. I created amplicon sequence variants doing this. Looking at the results, on the x-axis, we have our Indian River Lagoon isolate and our Old Tampa Bay isolate in yellow. On the y-axis, we have our Shannon diversity index. Our Indian River Lagoon isolate had a higher diversity, and there was a distinguishment between the free-living and particle-associated bacterial communities. However, our Old Tampa Bay isolate did not have that distinguishment and was very low diversity. Looking at the relative abundance, again, the Indian River Lagoon had a very high diversity microbial community associated with it. However, the Old Tampa Bay isolate did not have that distinguishment, or did not have a high diversity microbial community. In fact, only 9 amplicon sequence variants were detected in each of the particle associated, which is seen on the top panel, and the free living seen on the bottom panel. And these amplicon sequence variants were bacterial communities that were sediment and soil associated. I then want to determine if pyridinium can recruit bacteria from different sources to supplement B12. So sources of bacteria were collected from our Indian River Lagoon culture, which we just found out has a very high diversity microbial community associated with it, Old Tampa Bay surface seawater, and Indian River Lagoon surface seawater. I took each of these sources and I filtered them to separate the eukaryotic cells from the bacteria to isolate that bacteria. I inoculated the bacteria in our Old Tampa Bay isolate and allowed it to grow for approximately 2 weeks for recruitment. I inoculated each of those treatments containing our Old Tampa Bay isolate and that bacteria into the 2 vitamin treatments again. I measured chlorophyll A fluorescence every 48 hours, and at exponential growth, I performed a serial transfer again. After serial transfer 1, I filtered each of the triplicates using the same process as before for RNA extractions, and this time I did nanopore long read sequencing, as this gave us a higher turnaround time and a higher taxonomic resolution of the bacterial communities that were recruited. From the results, on the right side, we have the microbial communities recruited from our Indian River Lagoon culture, and then on the left side, we have our microbial communities recruited from our Old Tampa Bay seawater. The top panel is our particle-associated bacterial communities, and the bottom panel is our free-living bacterial communities. And then on the x-axis, we have our true treatments that the pyridinium and that bacteria was grown in. And these bacterial communities, again, were sediment and soil associated. And there was a difference in the particle-associated and free-living bacterial communities that were recruited. If we go back to our initial plot, our Old Tampa Bay isolate did not have that distinguishment between particle-associated and free-living bacterial communities that were recruited. So using the chlorophyll A fluorescence, I was able to create growth curves. And again, with the Indian River Lagoon culture in our Old Tampa Bay water bacteria, we can see that it did not supplement sufficient amounts of B12 to support growth. However, our Indian River Lagoon water bacteria may be able to supply sufficient amount of B12 to support growth. So my next steps for this project is to characterize the bacterial communities that were recruited from the Indian River Lagoon culture or Indian River Lagoon water. And then I will characterize the bacterial communities that were initially present in our Indian River Lagoon water and Old Tampa Bay seawater to see if pyridinium selectively chose bacteria into its phycosphere. Some key takeaways from this project is that pyridinium requires B12 for growth. Pyridinium recruited bacteria from Indian River Lagoon culture, Old Tampa Bay water, and Indian River Lagoon water. Most likely, we will still have some work to do. And bacteria from Indian River Lagoon culture and Old Tampa Bay water did not supply B12 sufficiently to support growth. But bacteria from our Indian River Lagoon water may be able to supply enough B12 to support growth. But is it enough to support bloom levels? I would like to thank my members of the MECO lab and my friends from the FWC for loaning me my cultures to work with. And then I would also like to thank my funding. And I'd also like to thank my pyridinium for surviving 2 hurricanes without any power. So thank you. Questions? Yes.
So obviously, the two populations of Pyridinium in Indian River Lagoon and Old Tampa Bay respond differently. Bacteria. Well, the Indian River Lagoon water has not been tested. Let's say it responds differently. So the question is, if somebody brings a bucket of Indian River Lagoon water to Tampa Bay, what will happen? Would that be invasive and to cause the whole population in Tampa Bay respond to change their response?
I'm not sure. That's a really good question. It does seem like the pyridinium, there's some bacterial community possibly in the Indian River Lagoon that is supplying that B12 that's not in Old Tampa Bay. So that is the big question, is what is this bacterial community? So that's what my next steps are. I'm going to look at those bacterial communities that are in the Indian River Lagoon to see if it's a bacterial community that's different than what's in Old Tampa Bay, and that could possibly be the source of B12, helping them to grow.
Thank you very much.
Yeah, that was great talk. Curious, is there any, do we know what the B12 levels are, like the concentrations of B12 in the 2 different waters, that maybe the Indian River Lagoon has lower B12 there, and thus it has grown a community of organisms that can make it themselves, whereas Tampa Bay water or Old Tampa Bay has higher B12, and so there's no sort of incentive for a bacterial community that itself.
Yeah, that's also a really good question. I have not been able to find levels of B12 in both. I've searched far and wide, but I feel like that's a good, there's a need for that, for us to test. If this is happening, we need to test the waters and see what it is and sources of B12. If it's not coming from bacteria in Old Tampa Bay, where is it coming from? Because it needs it to grow.
Lovely. Yes, please come get feedback forms if you're interested in filling them out. At the end, yes.
You bring some.
Yeah. Yeah, we can test.
So to change slides, so I just click the mouse.
All right, so our next oral presenter is Bella Ayanada, who is a master's student with Dr. Luther and Dr. Schwing, and she will be talking to us about spatiotemporal characterization of sedimentary phytopigments.
Take it away.
All right. Hi again. I'm still the other Bella, and I'll be presenting on some of my undergraduate research that I'll be building on for my master's thesis. So once again, it's spatiotemporal characterization of sedimentary phytopigments in the southeastern Clarion-Clipperton zone with these collaborators listed here. And forgive me if I have to take water breaks. I'm recovering from an illness. Sorry. So for background, what are phytopigments? For a biological standpoint, phytopigments are within our photosynthesizers used to capture light for photosynthesis. So I'll be talking about chlorophyll A and phaeophytin, which is the degraded version of that. So in sedimentary analysis, chlorophyll A usually indicates labile organic matter. And on the left here is a chlorophyll A molecule. I tried to highlight in red that central magnesium ion that when it degrades via digestion, cell lysis, or just plain degradation, it turns into phaeophytin here, which is on the right, and you notice it's empty here in the middle. So this is a graphic I made that shows kind of how phytodetritus makes it to the seafloor. So we start with our phytoplankton photosynthesizers here containing chlorophyll A. They'll either die off, they'll be grazed by phytoplankton. Phytoplankton are pretty sloppy grazers, so they'll make particulate matter. They'll ingest organic matter, and that'll form macro aggregates and fall to the seafloor as marine snow. It'll be respired by bacteria on the way down, and zooplankton will eat it on the way down. So only about 1% of what's up here will make it to the seafloor, and I'm talking about the abyssal pelagic, which is about 4,000 plus meters deep. So then we have our phytodetritus feeding the seafloor. So the phytodetritus contains the chlorophyll A and the phaeophytin, and it's feeding our nematodes. I tried to draw some benthic thoriums. These were some of my friends' favorite species. I forget their names. Also, it'll feed microbial activities. We've got brittle stars and holothurians on the seafloor as well. So my phytodetritus also shares the seafloor with polymetallic nodules that we just learned about a couple minutes ago. They contain critical metals, and we learned that mineral extraction will involve removing the top layer of sediment. So I'm also looking at the Clarion-Clipperton zone. I don't know if you guys can see it circled here in the Pacific. I'm looking at the southeasternmost part right here. This is NORAD. And once again, a nodule removal experiment occurred. This is from my friend Jaden Hyman, who published kind of an interesting graphic showing the collector plume, and then it'll create tracks, which is what I'm concerned with. Okay. So the goals of my study were to set baselines for natural variability of phytopigments in the sediment in NORAD, and to compare those results to other areas of the Clarion-Clipperton zone in the globe, and then also to analyze the change in concentrations of phytopigments after the nodule removal. Sorry, water break. So for sampling, we have 5 different cruises that are compiled into this data set. Here's the table that shows that. We had a test mining area here, and then a preservation area up here. And then down here are the tracks. We already saw this. So we used multi-cores aboard a couple different research vessels and extruded cores and sliced them into 1 centimeter increments from 0 to 5 centimeters, and then froze the cores at -20 degrees in the dark until extraction on land. And then I used Robert Navarro's 2010 methods to do acetone extractions and analyze them with a spectrofluorometer here on campus. And then I use these equations listed here. I don't really have time to go through all of it, but to take my counts detected on the spectrofluorometer and turn that into concentrations within the sediment. So for results, we established the natural spatial and temporal variability of chlorophyll A from 0 to 1 centimeters of sediment, phaeophytin, and then CPE, which is the combination of chlorophyll and phaeophytin. So these, the black dots signify a significant difference in the preservation area in 2020 and the test area in 2022. As well as a significant variability for chlorophyll A in the test area in 2021 and 2022. Also, the lowest quality, so the lowest amount of chlorophyll A compared to phaeophytin and that total amount of phytopigments was also detected in the baseline 2022 campaign. So these results suggest interannual variability in deposition and quality. It could be due to changes in POC flux, particulate organic carbon flux, or sea surface productivity. That's kind of a whole other animal. So my results here, this is my average CPE for two of my baseline values, compared to other baseline values in other parts of the Clarion-Clipperton zone on a gradient moving east. So we see that it kind of compares pretty well to those other areas. And also you see a gradient moving west, a decrease. That's consistent with Jessica Volz's 2018 paper. She stated that in the CCV, POC flux decreases moving west, which is pretty consistent with that equatorial upwelling zone, as well as a decrease in sea surface chlorophyll A. I don't know if you guys can really see it, but this is lighter purple here, which is like barely any sea surface chlorophyll A versus the darker purple, which is at least a little bit more. And then my values for baseline chlorophyll A from 0 to 1 centimeters of sediment is here in the yellow. So right here, it compares well to the Porcupine abyssal plain, which is about 0.04 micrograms per grams up here, in a similar depth and depositional environment. So those compare well. And then these two studies found that chlorophyll A will increase with decreasing depth, which makes sense. And then versus like areas like riverbeds of Spain, super shallow, you've got super high deposition of chlorophyll A. So here's that key figure again. We see baseline values in the yellow here, very variable. And then inside the tracks, we have very low chlorophyll A, all the way up to about 200 meters or zone 2 from the track area. We see evidence of organic matter deposition. Sorry, you can't see. And that's also here about 200 meters from the tracks. And this value far exceeds the baseline value only a month prior. And then we have our control values here in the red. So we can see that comparison there. Definitely evidence of organic matter loading here. And then also evidence of organic matter removal here. And this is kind of an image of what it looks like. This is from Von Ampe 2020, this image here, but just to give you a visual. So we've kind of constrained a zone of influence of organic matter redeposition after the test. Also downcore, this is kind of the what we would expect a downcore profile to look like from 0 to 5 centimeters of sediment. Sorry, excuse me. So we have a downcore decrease. The orange is chlorophyll A, the blue is phaeophytin. So that's consistent with consumption and degradation. But then we have our post collection downcore profile here. This is the control area versus that site 200 meters from the tracks. You can see obvious loading of organic matter here down to the 2 to 3 centimeter layer. You also have this is inside the tracks. You can see a deep scouring depth there down to 5 centimeters. And then versus in between the tracks, a little bit of organic matter loading compared to a baseline value within where the tracks would eventually be exactly a month prior. So a year later, this has decreased here. This is that area that had that organic matter loading. Probably some grazing occurred there by your phytodetritus grazers. But overall, regular spatial homogeneity here compared to our control values that were not modeled to be affected by the plume. So implications, a previous study in the Peru Basin by Van Afme didn't see a whole lot of pigment or microbial activity return before the test values in 26 years. So this is a little bit consistent with that on a very small scale. So obviously biota depends a lot on phytodetritus. This is a figure cut off here from Jeff Drazen, I think 2008. So you see phytodetritus feeds are sedentary protists again, are meiofauna, generalists, holothurians, brittle stars. So the implications for this study, we showed organic matter removal in one location and loading in another location and likely some grazing there. So this data is under review for publication. I'm also going to compare it to data that I hopefully gather from the Penryn Basin of the Cook Islands, EEZ here. And I also want to expand on that and include parameters like stable carbon and nitrogen isotopes, percent total nitrogen and total organic carbon, and then the ratio of those to each other. Because I want to understand the organic matter source, degradation, the sedimentary carbon stock in this area here. Sorry, I forgot to put that out. I also kind of want to look at the influence of deep water circulation on the organic matter composition, if there is any on a small scale in this area here. And in conclusion, sorry, I'm losing my voice. I established natural spatiotemporal variability of my sedimentary phytopigments in NORI-D, and this is comparable to other areas in the Clarion-Clipperton zone and the world. Also, we demonstrated a zone of influence of redeposition of labile organic matter after the nodule removal test. Also, future studies will incorporate additional parameters in a different area of the Pacific that is also of interest for nodule removal. So I'd like to thank all of these people, especially Dr. Schwing, for trusting me with this project. I think we're coming up on 4 years ago that I started this, and being a great mentor, and also Dr. Luther for being a great mentor and taking me on here as a student. Yes.
Any significance to those particular regions, why they were chosen?
I didn't have a lot of control over the experimental design, but what I understand is that there might be a less dense nodule composition in the preservation area that was like far out to the east within the NORI-D area. And also, I think part of the ISA guidelines are to make it a point to have a preservation area that won't be touched by any sort of extraction activity. I don't know exactly what their thought process was with like that specific region, besides just having like an area to control or like compare it to. But that region was only sampled in the first 2 baseline campaigns. Any more questions? We have time for one more. Right. Thank you.
All right, our next oral presenter is Orion Sharton who is a master's student with Dr. Liu, and he will be talking to us about the development of East Florida Coastal Ocean Model. Take it away.
All right, everybody. As Bella said, my name is Orion. I'm a master's student with Dr. Liu in my 2nd year, and I'll be presenting some preliminary research on my thesis topic, which is the development of the East Florida Coastal Ocean Model. So to start a little outline, we're going to talk about why we undertook this study, the benefit of doing this kind of work and using numerical modeling as a tool, how we created the grid, the forcing we applied, and then our preliminary results looking at water levels and some tidal characteristics. And then we'll discuss where we're doing well, where we need to improve, and some future work we'll be working on. So as we all know, living in Florida, it's a densely populated state with a lot of coastlines and a lot of complicated geometry, especially on the East Coast. There's a lot of low-lying marsh flats and estuary systems that are threatened due to climate change, anthropogenic input, and of course, overdevelopment. NOAA has prioritized developing a lot of high-resolution models in all areas of the coastal U.S., and the East Coast of Florida is one where they currently don't have an unstructured model running, so this is of importance to them as well. So we had to choose our model, first of all, to figure out how we're going to take this study. And there's been a lot of previous work with the WFCOM for these kind of, or the, sorry, FECOM for this kind of work. It's an unstructured triangular grid that's comprised of unoverlapping triangular elements. So it makes it really easy to characterize a complex geometry over the structured grids that have been used in the past when computational power wasn't as great. Downscales from the deep ocean across the shelf and into the estuary. It's a 3D density independent model, and it's proven effective for both local and regional applications. Weisberg and Co. have made models on the west coast of Florida, and Jing and Co., Dr. Chen and Co. have made models on the Tampa Bay, as well as Dr. Chen up in the northeast of the U.S. So it's very robust, and it's been used also in some local estuarine studies in the Satia River estuary, which is in Georgia area. So on the right side of the screen, you'll see our full model domain. It extends from Florida Bay in the south all the way up to Jekyll Island, Georgia in the north, which encompasses the whole east coast of Florida. It runs along approximately 800-meter isobath. Mesh generation, bathymetry interpolation was all done in SMS software. The grid contains about 124,000 nodes and about 240 elements, 240,000 elements. Resolution varies from 3 kilometers at the open boundary all the way up to about 20 meters in the estuaries and inlets. And you can see this as we move to the center panel on the left panel. That's the St. John River's entrance, which has a resolution about 20 to 25 meters at the entrance. The red dots are the tide gauges. 7 are operated by NOAA. They provide water levels, and there are some ADCP current data over there as well. But for this first part, we'll just be looking at the water levels from those tide gauges compared against our modeled data. So the forcing applied, first of all, for our surface of forcing, we apply North American mesoscale model, which implies surface winds and heat fluxes. And we use sea surface temperature data assimilation through outputs from OSTIA, which is a 6-kilometer grid, and river discharge from USGS, and then one-way nesting with the global structured HICOM. We provide 3D velocity, temperature, salinity, and sea surface height from the HICOM interpolated onto our open boundary. And then we do include tidal harmonics and amplitudes from the TPXO global tidal model. So as I said, we're going to talk about some results of our water level comparisons. We compare a month of data for the first start here just to see how we're doing. 7 NOAA tide gauges, like I said. Harmonic analysis is done through the T-Tide program. And then we also use Wilmot Skill Score as a way to verify how well our model is doing. 1 would be a perfect agreement. 0 would mean terrible agreement. Most of our scores are pretty high, and our RMSEs for our tidal ranges are pretty good. They range from about 5 to 7% for most of the tidal ranges, with the exception of Dames Point, which we'll talk about later. So here's just the visual representation of the month-long data. The x-axis is the month of November from the 1st to the 30th of 2024. The stations go from south to north. I know it's a little backwards, but I had it set that way, and I forgot to flip it. Sea levels on the left, and our RMSEs are set up on the right side. Observations in blue and the EFCOM in red. So you can see visually it looks pretty encouraging. There are a couple points that you see in the Dames Point center section. We go through the king tide cycle, we don't quite represent it. And then so, visual is always great, but we need to kind of see where we're doing with our harmonics, as those are very essential to most of the tidal structures in the estuaries. We only show 4 stations here because the other 3 not included look very similar to the ones they're located next to. As for the St. Johns River, it's right next to Fernandina Beach, and the structure of the constituent looks almost identical. So as you can see, most of our amplitudes are pretty good for our 8 major constituents. Of course, the 3 smaller S2, P1, and Q1 are very small amplitude. So for the next slide, we don't look at those. They do align decently well, but since our amplitudes are so small, we just focused on the ones that are major drivers of the water level variations. And so our constituent phases, or phases for our constituents agree pretty well. M2 is in red, N2 in green, K1 in blue, O1 in cyan, and S2 in purple. As we see, we have about a 20 degree RMSE for our phases, so we're getting them pretty close to right, but of course, we would love to improve on that. And so we've incorporated these. You'll probably see this slide similar to this a couple times a day. There's other people discussing similar work. But this is a new automated nowcast forecast system that the Ocean Circulation Lab has developed. It's a combination of our three operational models right now, TBCOM, WFCOM, and the EFCOM. We run those daily on the HPC system at Tampa. It's a 1-day hindcast and a 3.5-day forecast. And on the left side of the panel, you'll see our interactive map. If you click on one of those little red stations, a prediction will pop up like this, where blue is the NOAA tide gauge data, and then the red would be the prediction from the model, depending on which domain you're on. This one's obviously for the East Coast, so it's the FECOM. And you can scroll around. We have tide gauges located all around Florida with model observations or model simulations next to them. And so we give you 3.5 days, which is the red out. So overall, preliminary results for this, it is basically just a tidal model at the moment, but we are getting the tides pretty close to accurate. Low RMSEs around 10 centimeters, which for NOAA, they require 14 centimeters or less to transition to an operational model. So we're hitting a lot of their criteria currently. The one that we do very well at is Dames Point. Dames Point is located up the St. Johns River. It's around like the second crook, and it's right behind a little bridge or shoaling they have. So we need to improve the bathymetry clearly for St. John's, and then probably update it because it is a very active channel. So it probably sees a lot of dynamic movements in the bathymetry and dredging and whatnot that they do. So that's something that we need to probably constantly update our bathymetry for. And I would like to work on the bottom parameterization, improving that because we use a constant one for the grid. And obviously it's probably not constant all the way around our grid. Working to improve the accuracy of our offshore currents. And like I said, we are moving to a new bathymetry, which is the continuous updated coastal relief model from NOAA. It is their most accurate bathymetry they currently provide. It's been be automated since August of 2024. Obviously, we keep making improvements and updating the grid that we run it on, but it is part of our workflow currently. Like I said, our preliminary results are promising, at least for not knowing anything about unstructured modeling before I started this. I'm pretty content with where we are, but of course, always have to be ambitious on the future. So we're looking to, of course, analyze our tidal currents and our subtidal currents, do some low pass filtering and see what we're working with when we take the tides out. And then, of course, compare our temperature and salinity and the estuaries particularly and throughout our model domain. So in conclusion, we have developed a high resolution unstructured automated nowcast forecast system for a densely populated region that is in need of a model like this. It can provide crucial marine and hydrodynamic information for marine operations, navigation, both commercial and recreational. Some possible future applications that we're looking into that have been used in other domains. Pollutant tracking and forecasting, which was really important for the Piney Point spill happening here in Tampa Bay. Seagrass hacking and forecasting, which HIBO's done a lot of work in the Puerto Rico region for that. Storm surge and inundation, obviously living in a hurricane prone area, that is one of the highest priorities for numerical modeling work down here. And of course, interdisciplinary studies such as larval dispersion, with which Weisberg wrote a very nice paper about. And sediment transport is something I'm personally very interested about. The Port Everglades channel is set to undergo a massive expansion, and that'll obviously change their current structure of the channel and as well for the environmental impact of the sediment plumes. So that's where one of my important interests lies. I don't know if that'll end up in this degree or the next, but something I'm interested in. And with that, I'd like to acknowledge the NOAA Unified Forecast System Coastal Application Team. They helped a lot with the development, education, and training in FECOM. Like I said, when I started here, I'd never used an unstructured model, and through that help and a lot of help by my advisor and the lab as a whole, especially Seba and HIBO, they've really helped me get to where I am now. And of course, I'd like to acknowledge the funding from NOAA, Comet, Segura, and the Paul Giddings Memorial Fellowship. With that, I'll take any questions.
Tracking seagrass. Do you mean seaweed or did you mean fixed seagrass on the That's the habitat.
Sargassum.
Okay, good.
Yeah.
Thank
You can see my biology background is minor. Go ahead, Dr. Mice.
Between like the WFCOM and the EFCOM, you analyze what happens to things cross over the meshes.
It overlaps slightly, and we have looked at because that area is very challenging with the Florida Straits being such a narrow region. With high comms resolution, it is getting kind of hard to do that. So we have looked at applying the WFCOM output to the EFCOM using that as our forcing. We've seen some minor improvements, still something we're trying to figure out how to get that transition smoother. The currents for the WFCOM are better in that region at this moment, but so we're trying to figure out how we can more align the straits for that region. It's very narrow and it's obviously a huge impactor for the whole current structure of the domain. Juan?
Shows, 3.5 day forecast.
Yeah.
Is that like because of the uncertainty that the year, part of the picture or because of the days?
That was the workflow that we had going when I got here, and we kind of just kept it the same. That's what the other two models run off, and it's every day we update, so you get a new day ahead. So I think 3.5 is good because the weather patterns also, predicting farther than that is already uncertain. So I mean 3.5, I mean you can look at windy in 3.5 days from now. It's not going to be what you looked at in the forecast, so it's even more complicated here. Don?
So, the TP PXO type model is really designed for the open ocean. And it does reasonably well in the coastal ocean, but there's non-linear tides that start to come into effect in the coastal ocean. Have you guys started to look at the potential impact of these non-linear tides? Because I think that might be explaining some of the differences that you're seeing. I think that the Goddard model that's done by Richard Ray, the GOT, it may have some estimates of some of those major nonlinear tides. You might want to experiment with that as well.
I'm glad you brought that up because that's actually something I forgot to mention earlier. Our percent variance explained is only in the 80 to 90 range right now with those, but if we were to add those overtides, like you're saying, the shallow water constituents, MS4 and M4, those are anywhere from 5 to 8 centimeters in that region. So, I mean, when we add those two in, we get another 7% of our percent variance explained. So it would be, of course, we can't apply those at the open boundary, but I think a lot of that could come from better work with our bathymetry as we get, better generation of those.
You mentioned the currents. So one of the challenging things in this region is getting the phasing of the meanders. Like HICOM, there's a lot of room for improvement there. So it's a challenge to see if you guys can do better.
There's been some discussion of trying to incorporate some altimetry data assimilation and see if that would help us kind of locate it better. It's like you said, the meanders. I mean, I haven't looked at them exactly, but just looking at the general pattern, I know we don't have the Gulf Stream exactly right on where it is offshore. So something we're looking to try to improve upon.
It'd be very useful. Yes.
Yeah. We'll see what the computational power allows us for already. Yeah, we got time for another one if anyone else. Yes, Tony.
I think I noticed you presented the comparison of your results against previous model, current model used by others. For example, you said 10 centimeter would be square error on subpiece meets the NOAA criteria, right? It's above their criteria, so it's great. What about other models? How much improvement have you achieved? For example, Gulf of Mexico HICOM, global HICOM, and other models.
I haven't compared against the HICOM outputs from those areas, but I do know that NOAA runs a ROMS-based model up in the St. Johns River, and their RMSEs are in the 15 to 17 range for that area. So we are, not for Dame's Point, but for the entrance of the St. Johns, we do have better RMSEs, but as we get farther upstream, they deteriorate to about what the NOAA ROMS model has.
Thank you.
Bill?
Sure, you can just stick it here.
Sorry about that. I meant to.
That's fine.
Earlier, just. Fine, it's a last minute, that is fine.
No.
So that's gonna be. Thank you. All right.
All right, everyone. Please feel free to grab bagels and breakfast and coffee. We're gonna have a break in about a half hour, so thank you for bearing with us. But our next presenter is gonna be, hello, everybody. We're moving on, thank you. Our next presenter is gonna be Bradley Nemeth, who is a master's student with Doctor Who, and who will be talking to us about the characterization of trace metals via optical proxies. Thank you. Take it away.
Thank you very much, Will. Okay, so I'm going to be presenting on some preliminary research that I've done for my thesis topic. I'll also say that while this is not completely groundbreaking, this is still fairly novel in the sphere because the literature is incredibly limited to this. So without further ado. I'd first like to make a personal thank you to Dr. Frank Mueller-Carger as well as Dr. Kristen Buck for allowing me to use their MBON and NOAA AML data. Without this, I could not really do this project because when you're out on a cruise ship and you have optics measurements being taken, you have chemistry measurements being taken, very rarely do the two scientists ever talk to try to correlate their measurements. So I'm the one who's taking this mad task. So the purpose of the research, trace metals are notoriously hard to measure. There's a whole entire protocol, it's called the geotraces, where you have to have various specific protocols set up on a ship because most ships are metal. So my hope is to potentially use the in-water optical proxies such as CDOM, chlorophyll, detrital and backscatter, and try to see if there's any correlation between those and trace metal measurements that were taken during the AML cruises. Ultimately, why to do this? So maybe we can go from in-water optical proxies and blow it up to remote sensing, so we can kind of see if we see massive plumes out on the West Florida shelf, we can kind of say, hey, there might be some trace metals in there, because ultimately we want to know how, when red tide originates off on the shelf and kind of gets blown in, we want to see how they interact with those trace metals. So I'm going to give a quick little biochem geochemistry thing. First, why? Obviously, everyone here lives in Florida. There's been a lot of work done on red tide research, but we still really don't know what initiates them. There's been multiple papers done by Dr. Weisberg, as well as papers done by Dr. Gu, just trying to figure out, and other scientists trying to figure out where does red tide originate. So if we can kind of understand what their nutrient requirements are, or at least, you know, they go from essentially oligotrophic waters on the shelf, they get blown into the coast, they go from famine to feast. So if we can kind of characterize these trace metals and kind of see, you know, if there's any way we can see this via remote sensing, we can kind of understand, hey, if we see a lot of plumes and such on this Florida shelf, we can say, hey, that area is primed, and kind of help better understand how the red tides interact. So why are trace metals important to phytoplankton? Taking you back to some chem and bio-osh. Each trace metal has a particular requirement. Iron is good for nitrogen fixation as well as oxygen. Cobalt and copper are various use for proteins as well as vitamins within the cell. So, but only needed in trace amounts. So a lot of the trace metals that we look at are, you know, in the nanomolar region and such. So the cells only really need a little bit. So trying to understand those trace metals with optics also shows where they are important to the cell. Okay, so let's give a little optics. What does a satellite see? So this is an ERGB that was taken on January 25th. This is ERGB, so what normally people will see on satellites is you'll see those nice big rainbow pictures of chlorophyll where, you know, cooler colors, not a lot of chlorophyll, warmer colors, lots of chlorophyll. I just did some ERGB right here just to kind of say, hey, what does the satellite see? So, Up there in northern Florida, we can kind of see that as a CDOM plume. CDOM is, for those who don't know, it's color dissolved organic matter. Obviously, we have our satellites taken from the optics website. We see a little bit of a red tide patch down in South Florida. So if you overlay that with a chlorophyll, you'll see a lot of very warm colors there. And then just because we are in winter and we are in the dry season, we do have frontal systems that are coming through, and they does a lot of wind driven mixing. So you'll see resuspension on the shelf because that is very shallow water. So those really bright patches are non-algal particulates and suspended sediments and such. So you can kind of see. So those are kind of the proxies that I'm looking at. So background and methods. So our study region is mainly constrained. The data that I'm presenting here, I have the Florida Keys incorporated. Moving forward, I may just cut that because this area is very optically complex. You have really shallow water, light hits the bottom, bounce up. It makes it a pain to try to suss that out. So we might be constraining it to just south of Naples, but this area is mainly primed for red tide. We see it in the news all the time, fish kills impacting tourism, local health and such. So that's why we chose this study region. My criteria was essentially there was a chemistry measurement being taken, there was an optics measurement being taken, even if they were within the same day or the same time, that still says that they're sampling from the same water. So that can allow me to build reasonable relationships. And the outcome of using is just we're trying to understand how these optical properties may influence the trace metals. This is really work that we're trying to just characterize these metals based on optics, give it at least something. All right, so come with the journey on me on data. So I first did a correlation matrix just to kind of see, what each trace metal. This research, we're only looking at dissolved metals. There is also measurements that have total dissolved. The only difference between dissolved and total dissolved is dissolved, what passes through a 0.2 micron filter. Total dissolve is just literally the seawater that you take up when you sample. So you can kind of see that in the blue, that is all positive relations. Obviously, darker the blue, more of a positive relation. Lighter colors, not so much. But one thing I'd like to draw your attention to is you look at the chlorophyll, the detrital, the CDOM, and ADG. And ADG is just kind of that CDOM and detrital put together, because that's also something a satellite sees. We can see a lot of positive correlations with the various dissolvable trace metals. Well, what does that really mean? It means that those are metals that can be bound to various ligands or particles that, can be readily biouptake. So essentially, this is kind of what we're looking at is we're trying to see, hey, we have an optical proxy, we have this dissolved metal. Can we now see, hey, this is nutrients that are available for phytoplankton uptake? So, and then we can kind of see how there are some trace metals that are more, correlated to other trace metals. I haven't really done much exploration into this, but it is something that did stand out to me. So it is potentially, more relationships that can be built. So with the dissolved, as people know with trace metals, sometimes you can either deal with not a lot of trace metals, or you get really down into the fingerprint region. Limit of detection essentially means that there is something there, but it's just the instrument can't pick it up. So with this preliminary research, what we looked at is, I'm looking at CDOM because CDOM has siderophores and other ligands that can bind that iron to it, which means that it can be more bioavailable. And I essentially set up a trend line. These are based off of 4 M-bond cruises that we had dissolved metal data for. And I can kind of see that there is a nice relationship between CDOM and iron, which is obviously from ChemOsh, we would expect that the iron is binding to the ligands and such. So I was very pleased to see that. Another one that I thought was also really interesting was cadmium. So cadmium is a bit of an interesting trace metal. It can stand in as a substitute for zinc. If a phytoplankton cell does not have enough zinc, the literature that I've done research on shows that the phytoplankton will uptake that cadmium, and that cadmium will be a suitable replacement. So but this metric that I used was ADG. That's just the detrital measurement and the CDOM measurement. Detrital is after a filter pad analysis, all the pigments have been stripped. It's just kind of that remaining particulate matter on it. So that also shows a nice relationship. All right, and then I did a case study just because I kind of wanted to see. West Florida Shelf is a very dynamic system. I've read the Bob Weisberg papers and Dr. Liu papers and trying to understand just the physics out there, and it's pretty complex. So this one was a case study. It was in the dry season, it was winter, and it was December of 2020. So you kind of see it. So we have ADG on the West Florida Shelf. Obviously, the warmer colors are showing that more towards the coast, you have a higher signal. More towards off the coast, you have less of a signal. And it kind of agrees nicely with the chlorophyll. This is me just trying to understand what the optics are out there. And obviously you expect closer to the coastline, you have more CDOM, you have more inputs and such. So you're going to have a higher signal out there. As you go further off, more of the particulate goes out. There's not much as input because we're in the dry season. So you're going to have less of the signal out there. So now going to our trace metals, this two ones that on this case study stood out most to me was iron and copper. Obviously, we see that iron is very high towards the coast because we have Port Charlotte, we have the Caloosahatchee, so those are two potential point sources. Emily Hall did a wonderful paper, but it was with macronutrients, but the literature also supports that these estuaries are point sources for trace metals on the West Florida Shelf, and we can kind of see that copper is also a kind of showing the same behavior. I will also say with iron, we also do know that there's Saharan dust deposition. So if a frontal system comes through, kicks up that bottom sediment, they'll resuspend that dust back into the water column, and therefore makes the trace metals more available for uptake. So because we're trying to tie optics and chemistry, you have to do a thing called PCA. So I'm going to say this first. This is very rough data just because it has to take a really look at, but I'd like to at least show you that as we move further this way on principal component 1 axis, you're getting more towards your offshore. As we move this way, you're getting more to your inshore, and then seasonality on principal component axis 2. Spring and winter are more towards this way, and then your summer and fall are more towards that way, which that is showing that there is the shelf does go through a seasonal change where you have, during the fall and spring, you have, those are generally our transition periods. So they're moving from wet to dry, and where you're in the winter and then in this summer, you're kind of in that, really dry season and you're seeing the wet season. So that was something that stood out to me a lot. Just to kind of understand, because essentially what we're trying to do is we are trying to track water masses and such, just to try to see where those trace metal tracers are. So discussion. So there is a positive correlation with optical proxies. That did make me very happy, even though I was totally expecting to just say, nope, can't do it. Don't do this. PCA indicates that there is seasonality, which I'm very happy. More data are needed. So there's only a small thing of dissolved, but there's a higher of total dissolved. So we're going to kind of pivot from dissolved over to total dissolved. Next is we're wanting to break into separate years to provide better tempo and spatial resolution. We're essentially just trying to see, there was an event that happened. Do we see that there's, those events have some correlation to the trace metals out there? And then we're also trying to do backscatter. Cannizzaro et al. Made a very nice relationship between the detrital and backscatter, where they are very tightly correlated. But we've been trying to work out with some MBON in situ data, but we're kind of pivoting a little bit to see if we can go look at some satellite backscatter data and then reference that against the in situ just to see if we can trust the data. So I will open this up to questions.
Up with the idea to do this.
So Jennifer Cannizzaro, who's in our lab, she is kind of the point of contact that's working on the red tide projects and such. And she was looking at essentially the optical data, and she looked at some of the trace metal that Kristen Buck did, and she's like, wait a second, there's some nice happy overlap. So essentially it's another way to try to understand red tide and whatnot. So if we can kind of put this biogeochemical standpoint and with optics, it can provide a better way to look at that. So that's kind of how that idea came up. So it combines optics and chemistry, which I'm very happy about.
What you're-
I can take it out.
Let me inject it first. Oh. The big red?
Yeah.
Check.
Just
I want to get it right. Is this your first page? No. All right, so this will be our last oral presenter before our 15-minute break. Thank you for bearing with us. Make sure you get your raffle tickets from Delphina after this if you're interested in participating. And without further ado, our last speaker for this section will be Luis Sorinas, who is a PhD candidate with Dr. Liu and Weisberg, and will be talking to us about the heat flux exchange of the West Florida continental shelf. Take it away.
Hey everyone, so like Bella says, my name is Luis and I'm gonna be talking about heat fluxes, how they influence like water column temperature on the West Florida shelf after the passage of cold fronts and hurricanes. So a bit louder. Okay, can you hear me now? Yeah. Bit of a background of why are we doing this. So previous work by us in like 2023, chapter 1 of my dissertation. We did monthly mean climatologies. We basically look at the seasonal variations and net surface heat flux and how that impacts the seasonal variation of the water column temperature for the West Florida shelf. However, something that come out of this is like, well, we tend to see that these events are rarely gradual. They usually happen in short-lived kind of tense events where they remove large amounts of heat in a short period of time. So the idea with this, this will be chapter 3 of my dissertation, would be to focus on use the same methodology we use for chapter 1, but looking at different time scale, shorter time scale on the synoptic time scale, and look at cold fronts and tropical cyclones. A bit of the material, so we're going to be using 3 moorings, one on the 25-meter isobar, 2 on the 50-meter isobar. They all come from the Coastal Ocean Monitor and Prediction System that is maintained by the Ocean Circulation Lab. We're gonna be looking for the purpose of this presentation to 5 different tropical cyclones. We have Ian in red and Eta in like that brown kind of color. Those are hurricanes that when by the buoys, close to the buoys, one is Ian, we all remember, very strong. One is like a weak sort of storm. And then we're gonna also be looking at Wilma and Dennis, which are pretty interesting because they don't really like go through a shelf, but you still see an impact on the water column stratification due to these events. It's like very pretty, like kindergarten level plot. I'm proud of. Like we're gonna like walk you through like net heat flux real quick. So when we talk about net heat flux, there are 4 main components like that we consider. There's blue net short wave radiation, is radiation from the sun, and it's a main source of like input of heat into the ocean. Then we have net long wave radiation, which is understanding as like infrared radiation. The ocean release infrared radiation to the atmosphere, and the atmosphere like release infrared radiation to the ocean, and they balance. And usually this accounts for like a net loss of heat from the ocean. From now, we're gonna be referring to this when we add them together as a radiative portion of the net heat flux. Additionally, we have sensible and latent heat flux. So sensible heat flux is what you're used to when you put your hand on the stuff in the kitchen. That's what you feel. Latent heat flux happens through change of phases. So like when there's evaporation, this takes heat out of the ocean that is stored, and once condensation happens of this water vapor on the air, it releases. That's how the heat is released into the atmosphere. These 2 components we refer to as turbulent heat flux. And finally, I don't have time to go into this a lot. It's a complicated topically, but like there's many metrics that you can use to measure the heat content on the water column. Based on the literature, on the type of data we have, we consider like depth average temperature to be a good metric to measure how much heat is gained or lose by the ocean. Okay, so the outline of a presentation. Basically, we're gonna be looking at 3 different topics. First, we're gonna look at surface heat flux and the different mechanisms between cold fronts and hurricanes. Then we're gonna look at how this influences water column temperature, and we're gonna use a 1-dimensional temperature equation to try to like account for like some the processes that we don't really can see with this data. And finally, we're gonna look at this particular event where like positive, we call the positive latent heat flux events, where the ocean actually gains heat from the latent heat flux instead of losing. So first, this is a busy picture, but like stay with me. I'm gonna walk you through this. So this is only for C10. What we see there is like a clear sky radiation in red, which is only like the radiation from the sun that reaches the top of the atmosphere. And then in blue, we have natural radiation, which is what makes it to the surface of the ocean. So the difference in between the red line and the blue line is a portion of the radiation that is absorbed by the atmosphere that we see on this purple like second plot. The main point here is like when a hurricane goes by, like one of the buoys or in general the shelf, there's a significant I'm sorry. With the you don't see there's a banner on the bottom where you can see the hurricanes. Unfortunately, we don't see it here. So this is for hurricanes Eta and Sally. Like from now, like vertical red dash lines are hurricanes, vertical blue dash lines are cold fronts. So the one right here, that's Sally. This one is Eta. And what you see on the top picture is that there's a lot of there's a significant reduction in incoming shoal wave radiation. When a hurricane goes by that you don't see in either of the 5 cold fronts. These are case studies, but we see it like we look at like 18 different case studies, and like this is a pretty consistent result. Then we have like the other component on the radiated flux is the net long wave radiation. Based on time, I don't want to go too much into detail, but the main point is like cold front bring like dry air. The dry air is gonna like decrease the net long wave radiation. I'm sorry, and then like they're gonna decrease relative humidity, like water vapor content on the atmosphere, and therefore like incoming shoal wave radiation. Hurricanes usually are associated with like a lot of like convective clouds, so increase incoming shoal wave long wave radiation, and they result in like in a net decrease of long wave radiation. Bottom line here of these pictures of this plot is like the decreases on shoal wave radiation is much larger on the tropical cyclones. Now let's gonna move to the turbulent portion of the net heat flux. We have latent and sensible heat flux. We add them together. We have the turbulent component of the heat flux. Now turbulent heat is mainly like controlled by two variables. We have winds and we have like the air sea gradients in temperature and relative and like specific humidity. So if we look at the winds, we see there's a different mechanism. So for hurricanes, like the turbulent heat loss is mostly driven by strong winds and weaker like air sea gradients. For cold fronts, like winds are relatively weak in the order of 10 meters per second, and instead, these weak winds are compensated by very strong like air sea gradients in temperature and humidity. Additionally, like as a caveat, like cold fronts typically tend to have smaller peaks, but they last for longer. So over a period of time, like they tend to be more efficient in removing heat from the ocean. Let's move to the water column temperature and the one-dimensional temperature equation. So this is for Hurricane Ian. Again, I apologize about the banner. So what we see there is like temperature at marine C10, C12, and C13. This we see in all the moorings that there's like a mixing, the wind-induced mixing. So the water becomes, it doesn't matter, it erases certification because well mixed. Please focus on the black line. That's the depth average temperature. And what we see is like we usually see like a 2 to 3 degrees like decrease in what depth average temperature. Now we focus on C10. We kind of want to like separate what is causing this. So we use the one-dimensional temperature equation. So this here is the depth average temperature at C10, and with this equation, we can kind of like account for how much of that can be explained by surface heat fluxes alone. And we see that for this case, most of the variation comes from surface heat fluxes. Unfortunately, like when you're working with buoy data, you cannot really account for advection and other like oceanic processes. So what we do is that we subtract like the real variation from what we can explain from surface fluxes. And the result that we're going to refer to as ocean dynamics includes advection, welling, and of course, like errors and other things. For this case, it didn't play a significant role. And this is not out of all the case studies we see. This didn't really happen a lot. There's another 2 more like cases that we have. This is Hurricane 2005 during Wilma first and then Katrina. We see the certification was really strong at C10 and C12. There's not enough data at C13. We see like a decrease in temperature too, but this one has a different pattern that is interesting. So the ocean dynamics control most of the variation in temperature. If you look like here compared to here, with only a minor contribution for surface heat fluxes. And this is pretty typical of storms that don't really like hit the shelf. They like go around. And so like the cloud and the influence of the on the air sea flux is not there, but they still impacted the certification on the shelf. And finally, this is Hurricane Hermine in 2016, I think. And this is interesting. If you look at C10, the black line after the pass of the hurricane, and there's instead of having like a loose of like depth average temperature, the depth average temperature actually like it increases by like about a degree. This is typical of weaker storms. Due to mixing, you like increase the bottom temperature, and like this can increase the overall heat content of the atmosphere. Oh, sorry. There's like this happened in 2020. There are cases where like this can precondition the shelf, and there's a second storm coming like in the month or so afterward. This increases the potential for rapid intensification. Real quick for this one, this is another type of like scenario we see. Like there's like contracted influence, so like ocean processes tend to in this case like warm the ocean, but well, one. I think so.
Shut off all the wind. Sorry, Louis. It's still blinking. I gotta wait till it stops blinking. Turn it back on. You have to physically. We might have to, because otherwise it's gonna cycle down, so it's cooling off before it shuts off completely, and which prevents me from turning it on. Yeah.
Is the light on the
That's
projector?
solid. It's solid. That's a good sign. Now let me try this. Now it's blinking, turn back on. You might need to unplug it and plug it back in, my best guess. Let it get- And then plug it back in after it's done. There it goes. Yep, good work. Come on. I saw the light come on. Where isn't it?
It takes a little while for it to come on.
Projector's on. I see a light.
Yep. Just
But
be
it's not
patient.
coming. Let's just warm it up.
Sorry.
Little progressive maintenance.
Yeah.
Yeah, it has to reboot because it went to sleep. It's still waking up. Have any more coffee for the projector?
That's some cold water.
Yeah, that's a little better.
Oh my god. Yes, but boy. Yeah.
I've been
I've been at like, I don't think we're something that's a wife. But true, is that it, right?
They don't know.
Yeah.
Yeah, let's try it one more time. Your screen's still on, right?
Yeah.
Try it one more time. Display on. I see the light. I don't know why it's not coming through. Are you?
Do I have to do this?
Oh, you know what? Maybe you have to hit PC here. You're on PC, right?
Yeah. Okay.
Maybe it's just input.
There we go. All right. Yeah.
Display settings at the top.
Yeah, right.
Uh-huh.
They were still. That's the way. Well, finally, I found something.
Sorry for that. Okay, so real quick, like this storm is really cool because it increases the temperature of the heat content the heat content of the water column temperature. And this is typical of like weak storms. So finally, let me rush through this real quick. We're gonna look at like one particular case when let me get there. So when we think about latent heat flux, it's usually like the main source of like one of the main sources for like removal of heat from the ocean and like from like driving like hurricanes, and that's usually the case. But we this happens during Hurricane Eta in 2020, and like again the dashed line is where the hurricane happened, and what you see there is like there's couple of instances where like the latent heat flux became positive for like about 50 watts per square meter or so, which is not a lot, but it's pretty counterintuitive. We like look at the Saturday vapor pressure and the air partial vapor pressure, and what appears to be happening is that the air is saturated, the latent heat flux is shut down, and instead condensation happens, and it seems to be like releasing heat into the ocean. We don't have enough data about describe exactly what's happening, but if you look at the temperature, this is what happened during ETA. You're gonna have to believe me, there's a 0.2 degrees warming there. But actually, if we like zoom in, this are about 0.2 degrees. Again, this is not a lot of like temperature change, but for a hurricane, like I think this, like the point we're trying to make is hurricanes are not all the same. They're pretty different. Like they have like variations in between storms and also spatial variations. And for like purposes of like heat removal from the shelf and certification, cold fronts seem to be way more efficient and way more consistent in how they remove heat from the ocean. This is a lot of text, but the main like point here is like radiative fluxes mainly are affected through like the reduction in incoming short wave in tropical storms. This doesn't happen for cold fronts. For turbulent heat flux, there's like an interplay between air sea gradients and air sea gradients and winds. For like hurricanes, winds tend to be the main driver. For for like cold fronts, it's the gradients that are like basically driving the latent heat flux. All events resulting in like well mixed water column. There's a reduction in most cases of the depth average temperature. And finally, like when we look at like what's happening in the water column, there's for cold fronts both constructed influence for cold fronts from like ocean dynamics and surface heat flux, and this is not necessarily the case for tropical cyclones. Or how Bob put it, like although not intuitive, latent heat flux can actually warm the ocean. Questions? Alex.
Yeah, so storms, question here. Do you actually know what intensity they were when they passed over these areas like Eta? Because I was wondering, could the Eta intensification, depending on how strong Eta was when it passed over the shelf?
So like C10 is right about here. So actually like so I'm putting every 6 hours, so you don't really see it, but like right around here, it re-intensified, went back into a hurricane for a little bit.
So what I was wondering is, could there potentially be any sort of impact where maybe when you have that warming in the water, there's somehow be some relation to any existing storm eye?
It could be. That's something like I haven't looked at, like, but like that's a fair question. I'm gonna look at it. I'll tell you Monday.
I'm gonna take one more question.
Oh, go ahead. Whoever else said their hand.
I don't.
Oh, there is nobody? Okay, then I'll ask a question. Why don't the storms all pass through Key West? No, seriously, my question is, what is your uncertainty of the temperature? Because you're going from 0.2 to 4 degrees. What is your actual uncertainty?
So the uncertainty for the heat fluxes in particular is significantly larger, especially for hurricanes. For the cold, like when you have winds higher than 20 meters per second, there's a lot of uncertainty. For the temperature, like it's pretty accurate. We tend to calibrate the instruments, and the 0.2 degrees for the, I don't know, that's a question probably for Gabe, but it's like the 0.01 if yeah. So like we're like way over that margin. All right. Thank you.
All right, everyone, we're gonna break for about a 10-minute break to go to the bathroom, get refreshments, and we'll be back for the second section.
Do we have the laser? I know, that's
Exactly what I.
Good.
Take that.
This one, right?
Yeah. But, yeah. It was good to do this. To do that. Yeah. But why do we see this?
There, you can change, yeah.
No. That is good.
No, now it's good. No.
Yeah, what is it? And now?
So we need to stop sharing, maybe, and we can.
No, no, no. Wait, can we? No, that's good. Yeah, but it's not sharing anymore, so.
Yeah, like.
It's not going to.
We need to share the window, not the screen. Yeah.
Oh, it's there on the right.
Oh, sorry. So here you need to share. Window.
This one? Window, you said?
Yeah. So when we share the, you only see the power button, not the. It's a window.
Thank you.
Ah, good. Very good.
When you share the screen, we are seeing everything, what you are doing.
And that's good because here we see this, it's not good. But here we don't see it. It's great. Because some presentation we cannot see there and we cannot see the bar. That's good.
Let's make it easy. Off sharing.
Like there is GFS.
Okay.
There's names.
Okay, I will change this.
Okay. You can put the.
Yeah.
I need to edit mine real quick. I found it typing. Yeah, you can work on it. There's also our. This folder and your number.
Replace.
You want to? I'm making the slides. I don't have the presenter, then I can make 10 less. Okay.
Yeah, I think, yeah.
Sure, it could be as short.
Sure. Good. Yeah.
Thank you.
I just.
I would like I'll get there.
Oh, that might have to, yeah. Yeah.
just about like the string on it.
What?
Yeah. You're gonna die. You're gonna die. You're not. Yes, I am. Can I not? Like, what happened here? Just run away. Yeah, barely. And then everyone's like. What happened? I think actually, Drew, I'm gonna drop out. Are you next? I'm gonna drop out. No, you're not. No, you're not. Yeah, we're gonna be fine. The projector's not gonna turn off. No, completely different. I can do this. Put her away.
So like representation, but mine is PDF. You guys, right? Like just saying, okay. This is like square. Ah, that's fine. So you just have to like. How do I put it in this one? If you go down, full screen open. Hey, everybody, perfect. It's not showing. We need to speak another, like maybe. Okay, yeah, that makes sense. Okay, you guys are doing great.
Thank you.
Good job.
Okay, so that'll be. That looks good. Okay.
Thank you.
Yes, no, thank you.
Now can you check, get Mulligan? I put my flashback.
Yeah, I have it. So this one.
Yeah, there's second
next time.
PowerPoint.
Next thing?
Yeah, let me.
Yeah, you need to just.
The next one will be the doctor students from. It's okay.
This one, the final.
Yeah, like that in the flashback.
Yeah.
Updated.
Okay, so I'll eject your thing. Okay, I can remove it.
Hey.
That was busy.
What do? What if
When Luis was standing, they couldn't see.
Probably.
Some people like
Scared, like,
macronutrients.
oh, never mind, we are. Yeah, okay.
So, I don't know.
There you go. All right, everyone, we can try and find seats. We're about to move on to the next portion.
I thought maybe there was an
All right, everyone, if everybody's getting settled in, we are getting ready to start our next portion of oral speakers. Just a reminder, after these 4 speakers, there will be a lunch break in the NSL lounge. So feel free to head over afterwards. Again, a reminder, we have physical copies of the review forms and get raffle tickets at the end of this section of talks. And our first speaker is Emma Graves, who is a PhD student with Dr. Chappell, and she will be talking to us about examining phytoplankton community composition in the Southern Ocean.
Hello, I'm going to be talking about some work that I've been doing looking at phytoplankton community composition in the Atlantic sector of the Southern Ocean. So to start, as we all know, oceanic phytoplankton are of global importance. They perform almost half of global primary production, despite accounting for only 1 to 2% of global plant biomass. Phytoplankton community composition is regulated by the biogeochemistry and physical conditions that affect primary productivity. In the Southern Ocean frontal zone, where large nutrient concentrations and stoichiometric gradients exist, phytoplankton community composition is particularly variable. Previous research has shown that fronts coincide with chlorophyll concentration peaks. In addition, many previous studies have explored phytoplankton community composition in various sectors of the Southern Ocean, but most have only used imaging or pigment-based methods alone, which tend to result in lower ranked taxonomic groupings or morphological groups. These studies have provided valuable evidence that phytoplankton communities correlate with nutrient and temperature salinity shifts in the Southern Ocean. Specifically for my study, I was looking at the Atlantic sector of the Southern Ocean, crossing through all of those fronts and zones, with the two specific research questions of how does phytoplankton community composition shift along the transect, and what environmental factors potentially control these shifts. Our main hypothesis was that regardless of the method used, changes in phytoplankton community composition will be associated with front end zone transitions. We used 3 different methods, sequencing, imaging, and pigment-based methods to analyze phytoplankton community composition. So starting with our sample collection, obviously we started with water, which has the phytoplankton in it. We have both surface samples from CTDs as well as continuous underway sampling. And we did 3 things with all of those water samples. First, we filtered about 5 liters of seawater onto 0.2 micron Cervex filters for our molecular samples. Then we also filtered about 2 liters of seawater onto 25 millimeter GFF filters for our pigment-based analysis. And then we pulled some whole seawater samples for that imaging analysis. To get a little bit more into the nitty-gritty of the methods, for our sequencing-based method, I did DNA RNA co-extraction and then PCR with both a universal 18S primer, which targets eukaryotes, and a universal 16S primer, which we use to target eukaryotic chloroplasts specifically. After processing, the sequences were clustered into operational taxonomic units with 99% similarity, or OT99s, and assigned to taxonomy. For the imaging-based method, we used a pulse shape recording and imaging flow cytometer, or the cytosense. The cells imaged were assigned to one cluster based off of their optical properties using CytoClus 4, and 2 measurements were taken for each cluster or group: biovolume per milliliter and cell abundances per milliliter. And then lastly, for the pigment-based method, those GFF filters were extracted in methanol, sonicated, and clarified. And then HPLC measurements were taken of 13 different pigments, which were used as Chemtax input to obtain the abundances of 7 distinct phytoplankton taxa. So I'm going to show you a couple very similar figures. What we're looking at here is the relative abundance of different taxa. On the left, we have the most southern zone, the marginal ice zone, and then we travel north all the way to, on the right, we have the subtropical zone, which is our most north zone. The 18S primer resulted in a lot more dinoflagellate sequences. That's that pink mysozoa. Our communities were dominated by those. This is unsurprising though, because 18S dinoflagellates carry multiple copies of their 18S gene, and so they do tend to swamp out other things when you're looking at 18S sequencing. Of note, we did see in our more southern samples organisms like Heterocapsa and Catoceros, which are known to do better in colder waters and have wider thermal tolerances. And then in our more northern fronts and zones, we have organisms like Warnoia and Carlodinium, which have more like tropical distributions. A similar figure, this is for the 16S data. We see that the diatom and haptophyte communities dominate a lot more than the dinoflagellates. So in our most southernmost region, marginal ice zone, it's dominated by diatoms, specifically high numbers of pseudonychia. So diatoms have that cellular requirement for silicate and are known to thrive in the high silicate environments off of the Antarctic ice shelf. We also see Phaeocystis dominating throughout the transect, and that has been shown in previous research to be a major player in the Southern Ocean phytoplankton communities. And then in our most northern regions, we see a lot of Pelagomonas, which are known to thrive in low iron environments like that northern region of the Southern Ocean. Moving into our imaging data, a little bit different than before. On top, we have the absolute abundance of our biomass, and then on the bottom, we have the relative abundance of the counts. As you can see, in that biomass, it is mainly made up of large nanoflagellates, which is unsurprising because large cells have more biomass. But mainly we can see that there's higher biomass overall in those more northern fronts and zones, which is consistent with prior findings. When we look at those count-based abundance measurements, we can see picoprokaryotes, what we assume to be cyanobacteria, are a lot higher in those more northern fronts and zones, which again falls in line with some previous studies. We also see pico eukaryotes increasing. We don't have any more information as to what exactly the pico eukaryotes are. We assume that those include like very small diatoms and presentophytes and coccolithophores. And so overall, this method has really low abundances of those specific taxa like diatoms and dinoflagellates and no coccolith or very low coccolithophores, no presentophytes, and so they're falling more into those morphological groups. Lastly, taking a look at our chemtax data, we see presentophytes are highly abundant throughout. That's a group that we had not seen in any of our other results. Dinoflagellates and haptophytes are also present throughout. There is that shift in more diatoms in the southern region that we saw previously in our other data associated with high silicate environments, and then a shift of some more cyanobacteria in the more northern regions that we saw in that cytosense data. So we saw that all of these different methods had very clearly different community compositions that were resulting, but we wanted to see how they all compared when we associated them with different environmental variables. So looking at these cat plots, we have the 18S and A, 16S and B, C is the imaging counts, and D is the chemtax. And we can see that these samples are grouping by front and zone. That's the color. And then we saw that as well in our R-squared values for front and zone. They all were pretty high, 0.6 to 0.7, meaning that front and zone is able to explain a lot of the variation that we see in our phytoplankton community compositions. We also see that communities from more northern samples across the board are associated with higher salinity and temperature, and communities from more southern samples are associated with higher nutrients. So I said that all of those methods had very different phytoplankton community compositions, but those direct comparisons between methods can actually be really, really difficult. For one, you probably noticed some have morphological groupings and others have taxonomic groupings. So cytosense uses more morphological groupings. Chemtax has higher ranking taxonomic groups, and sequencing can really get at those species level assignments. So for example, in cytosense, we had those nanoflagellate groups, and if those accounted for or were just dinoflagellates, that would lead the cytosense imaging data to look a lot more like that 18S sequencing data. Chemtax also follows similar proportions of dinoflagellates to diatoms as seen in the 18S primer, but it has a large presence of presentophytes throughout the transect. So the two most abundant presentophytes, Micromonas and Ostriococcus, are extremely, extremely small, 2 micrometers in diameter. So these probably would fall into that pico-eukaryote size fraction that we had in the cytosense data, and might also just, due to their extremely small size, have less 18S and 16S copies and be swamped out in that DNA. Overall, we also saw a lack of prokaryotes in the sequencing data. Both of our sequencing primers were targeting eukaryotes specifically, and so we don't have any of that data in our sequencing stuff. And from the cytosense and chemtax data, we could see that cyanobacteria were more abundant in the northern regions, so our sequencing data is missing that variability. Overall, it illustrates the importance of using multiple methods in tandem. Cytosense and chemtax-based measurements are more cost-effective ways to gain a broader level understanding, but sequencing allows us to get at a more specific taxonomic level. So in conclusion, our sequencing, imaging, and pigment-based methods all had differing resulting phytoplankton community compositions. They all have their pros and cons, all those methods, but regardless of method, the conclusions are similar. Front and zone has strong correlations with phytoplankton community composition. In terms of future work, we're also exploring metatranscriptomics to look at what the phytoplankton are doing in those areas. And then I'm working on another project to explore the community composition relationship with front and zone in other sectors of the Southern Ocean. So I'll take questions now. Yes.
Really good job. I enjoyed your talk. And I was just wondering if your pigment-based methods with the HPLC were used in-house, or if you did that.
That was not done in-house. I'm not exactly sure where we got that data from. It came from a collaborator, Tommy. I don't know where he's from.
Came from
Yeah, so they just sent me the pigment data and then I ran the chemtax on it. But it was not done in-house. I don't actually know where he did that. Yes.
I was wondering how you define your fronts. Were they from satellite imagery or were they from institute?
Yeah, it was defined from data on the cruise. So we didn't have any like deep continuous data. So it was defined using surface data, which I know is sometimes not exactly the best way of defining those fronts. But yeah, it was done on the cruise. Yes.
One of the main objectives of the NASA PACE mission is to map phytoplankton community composition at a global scale.
Mhm.
So I would encourage you look into that,
Mhm.
compare your results
Mhm.
against a large picture or a more temporal change.
We have time for one more?
Sure.
Okay.
The question. The question is
Was that
simply,
relevant? Okay.
why would it say that the not being able to detect fronts from the ship is any worse or better than remote sensing? Have you tried comparing the two and seeing what might work best?
Like detecting the fronts, or are we talking about like phytoplankton, looking at remote sensing with
Question
phyto?
earlier about the fronts. Was that a question about the fronts?
Yeah.
And you said we were detecting it from the ship instead of remote sensing, and I was just curious, why is the ship detection not a good way to go?
From what I understand about fronts and the intermediate zones, the remote sensing is also not necessarily going to be the best option, because you want data from depth. So you want to be able to look over the water column. Most of like frontal definitions are done at like 100-kilometer depth or something. So
I would think that the ship observations might actually be better than the remote sensing. Honestly. Yeah. And my colleagues.
it's a touchy subject.
Whispering my ear.
our understanding from chatting Don who's thought about the Southern Ocean fronts is that if we had had very high resolution CTVs through that, that would be great. But what most of what Emma has for her data is that we didn't stop the ship, we sampled while underway. So there aren't
underway
Yeah, it's just surface data. We don't have any data at depth to
CTVs
like.
on the northern transect, but they were not a high resolution that done actually as opposed to what we did. So
So our frontal definitions are like a surface manifestation estimate of the fronts.
That helps clear.
Yes. We're good?
All right.
I both. Do you have the laser plans tomorrow?
I don't think we do.
Oh, okay.
You can use, if you move the mouse, it should show up on the screen.
Yeah, I tried.
Feedback forms? Anybody need feedback forms? As Delphina makes the rounds. All right, our next speaker is Haibo Su, who is a postdoc with Dr. Liu and will be talking to us about storm surge and coastal inundation nowcast/forecasts during our most recent hurricanes, Helene and Milton. Take it away.
Hello, everyone. Thank you for being here. My name is Haibo Xu. I'm a postdoc from Ocean Circulation Lab. Today, I'm presenting storm surge and non-cast coastal inundation nowcast forecast products we developed and used in response to Hurricane Helene and Milton's. As we all know, the West Florida shelf has been hit badly by three major hurricanes in 2024, Debbie, Helene, and Milton's. Among those, Debbie and the Milton's occurred only 2 weeks away, but they represent the two different cases of storm surge for Tampa Bay. Recall the hurricanes occurred in recent years. The Hurricane Helene set the new record on water level and surge height with 7.2 feet and 7.8 feet representatively. The Helene brought up the waters out from outside, but the Milton sucked water out of from the Tampa Bay. Those events threaten the public safeties and the infrastructures that make the timely forecast needed. The work I'm presenting here is not only provides the timely forecast in advance, in a couple of days in advance, but also have the implications on the coastal ecosystem issues, which could caused by the algae bloom, wastewater, sediment transport, and so on. This is a snapshot of the storm surge forecast models we're using. This product has been published before the hurricane seasons in 2024. It is the combinations of observation asset, including the tide gauge stations along the Florida coast and the three unstructured grade coastal circulation models, WFCOM, TPCOM, and EFCOM. All the models will provide us 1 day high cast and 3.5 days forecast. Those are match grade as from the models I just mentioned. The WFCOM is overlying on the West Florida shelf, extending from Louisiana coast to the Florida Keys, arching in the middles. The TPCOM is directly nested into the WFCOM, but has a higher resolutions. The AFCOM is concentrating on the eastern shelf with the slight overlapping areas with WFCOM around the Miami coast. The interface of our products is very user-friendly and practical. They are featured with zoom in and zoom out, whatever you want with your mouse. And clicking on any stations you're interested in could pop up the sea level movies like that with observations in blue and models data in red. I believe Oren has been provided a lot of details already. So here's I will put some examples for sea level product at Santa Keys, Mayport, and St. Pete's from 3 different models there. In case of the Hurricane Helene's, our storm surge products has been simulated the entire course of the Helene's. And we made a couple of comparisons between WFCOM models observations and another models operated by NOAA. The forecast of 5 each days leading to the landfalls are presenting here. The September 24, 25 until the 28th, with the figures we can show here is that the peak of the sea levels is underestimated by our models on the 4 days before the landfalls, but they got continue improve day by day. The simulations on the 25 is better. 24 simulation for 26 is much better until we got almost the perfect predictions. So overall, our models have the favorable agreements with observations. The forecasting capability got improved day by day. That is because the wind fields got updated. The more close to the landforce, the wind fields got more accurate. Let's we look at the performance of NOAA stoves. They're okay, but they're kind of underestimated for sure. The sea level for 2 more stations is percentage here. Besides the conclusions which we can draw from the previous slides, I also want to point out the semi-diurnal signals can be resolved very well by our models before the hurricanes here. And water level peaks can be also resolved by our models. We can see there's a competitive performance between those 2 models, but in the Tampa base, our models beat the stoves. The stoves cannot resolve any upper bay in this event. In case of the Hurricane Milton's, our results suggest there's no storm surge founded, but a slight negative storm surge. If you can say that this is the water step downs occurred around in the early morning of the October 10th, that cannot be resolved by both models. The reason for that is that the hurricane pathway is too close to those stations, and the wind force we're using has the interval of 3 hours. During the 3 hours, the wind can be changed dramatically in magnitude and speed. There's another story I would like to share here is that during the Miltons, our buildings and powers has been shut down by USF emergency office. So our models cannot be survived without any powers. Yeah, that's very. But the good thing is that the Dean, Tom, and the Garys got convinced by Dr. Jungang. They help us to reboot and put power back. So our models is able to keep running and archives all the valuable stuff like that. Thank you for the guys. And I hope in the futures, I'm not judging the decisions was made by the office, but I hope maybe in the futures our models can be used. Maybe I hope so. We're kind of off track. Let's go back to the main topic. There's another product I would like to present here is a coastal inundation nowcast forecast product. This models was created and initialized by the previous scholar, Dr. Jing Chen. To be clear, what exactly happened during the floodings, we extended the grade of TBCOM onto the land and put them together to have the total mesh grade of our models there. This models is featured by wetting and drying capabilities and initialized by the TBCOM on the ocean side. Model is not operational because it's not practical things in the Euro time in the normal time, but it can be run on demand. This model will provide the inundation forecast in 2 days in advance of landforce. Please keep in mind that during these studies, no rainfalls was included. Okay, so experimental products made realistic flooding simulations for Helene, as shown in those figures, as movies. Sorry, the the water portions has been removed or masked in black. So technically, the initial white point has been filtered out. All the data here is relative to the mean sea levels. I believe some of you or your friends or family's house got flooded. I'm so sorry about that, but the thing that how good our models is kind of depend on how severe floodings you got in your house. Sorry for that, but yeah, that's true. Yeah, when you feel better, let me know how good our models is. So this model, please remember this model is a experimental product. And this model need to be improved in the futures. Yeah, if you think there's any good from our product, please use it in the future or distribute it to your friends. So some takeaways. The unified storm surge nowcasting forecasting product has been developed and used in response to the hurricanes. All the models is configured to perform realistic simulations of model circulations and the water levels, which combine the time gauge observations to provide a 3 days high cast and 3.5 days forecast. Our models has been successful predicted the sea level set up and set down along the coast in advance, in 3 days advance. The result shows that favorable agreements with observations and overall advantage than the models NOAA operated. The Tampa Bay inundation forecast is also simulated during Helene, providing the forecast for 2 days in advance, identifies the areas with severe inundations. The last but not least, if you think any good of our models, again, please zoom up and distribute it to your friends and families. Any questions?
So just as a practical application, I woke up the morning of Helene and I looked out my window and I saw that the water was above the dock. And I knew from the model simulation that it was another 4.5 to 5 feet. So I called a few of my friends. I told them I'm going to leave. I left. My cars were fine. They didn't leave. It was a nice Mercedes Benz that got towed away and totaled. So the model actually did work where I live.
Thank you.
You mentioned that the wind field kept changing, and that would change your expectation for peak flooding or peak tide, water level of the tide gauges. The wind fields and the hurricane system, has the cone of uncertainty and something, and that's an assimilated projection.
Mhm.
I mean, can you include some level of error bars on your products so that you can say, if the storm track is going way north, then it's following, the European model, and that's more likely to give this type of response or something like that.
Yes, thank you. That is good suggestions. We will definitely we're gonna do these futures, but the difficulties that we're facing right now is that as long as we change the parameter like wind, we're gonna rerun the model again. I don't know how many times we have to rerun it to have the error bars like you described. But we are thinking to switch to another wind models with a higher tempo resolutions, which is HRR. They have like hourly wind. That can be really good practice. Thank you.
Got one more quick question.
This is probably a silly question, but great talk. Tampa campus, would it be possible to, like, in cases of storms that are bearing down in our general vicinity, could you offload that modeling work to Tampa campus? Because generally Tampa doesn't shut down unless the storm is like right on top of us, or would it be too much of a technological headache to try to move the computation of the modeling to Tampa temporarily and then move it back once the danger's passed?
Yeah, I think that is So that's a good suggestion. So and also we're thinking about that because our campus is more it's getting easier to be heated during the hurricanes there. So yeah, we're thinking about to move all the servers to Tampa's. Yeah, that's a good point. Thank you. Thank you.
All right, our next speaker is Felipe Stanchek, who is a PhD student with Dr. Chevanel, and who will be talking to us about testing alkanone unsaturation index proxies for paleotemperature reconstruction in the Southwest Atlantic.
Perfect. All right. All right, good morning, everyone. My name is Felipe. I'm a second year PhD student here at CMS with Dr. Emilia Chevanel. And today I'm not going to talk about Antarctica, clearly, but I'm still going to talk about a region in the Southern Hemisphere. So this project it's called Testing the Alkanone Saturation Index Proxy for Paleotemperature Reconstructions in the Southwest Atlantic. This is a product of the first chapter of my master's thesis back in Brazil, and hope you like it. So here's how I structured this presentation for today. So I'm going to start talking about what are alkanones and how we can use them in pollutionographic studies. Then I'm going to jump into the study area and how we collected samples for this project. Following, I'm going to describe what we found, what the alkanones are telling us in the South Brazilian Bight, and then the takeaway message. Oops. So alkanones. Alkanones are organic compounds produced by coccolithophores, mostly by the species Geophorocapsa huxley and oceanica. We can trace alkanones back in the geological record to the early Eocene when they were produced by species that no longer exist. But in the past 250,000 years, just these three species are the ones that produce them the most. Alkanones are long carbon chains. So as you can see in their structure right here. And in the late 80s, some studies found that the unsaturation level of this compound changes with temperature. So as temperature increases, we see more double bonds in this structure, which is these little things. And as temperature increases, we see more double bonds, which means that the unsaturation level is higher. So in 87, Kroll and Wickham developed a proxy that they call the Alkanone Unsaturation Index, or UK37, where they found this correlation between temperature and the value of this index. So as temperature increases, the value also gets higher. And then here I have a little cheat sheet. So temperature increases, we see more double bonds, which means that the unsaturation level is higher, which causes the UK37 values to be higher as well. And then in 88, Kroll and et al., they published the first calibration that converts this proxy into temperature. And this is a linear equation, and it's still one of the most widely used equations for paleotemperature reconstructions using this index. But there's been many other global equations that want to make this proxy more reliable when we use them for paleotemperature reconstructions. And here I have a few different approaches that these equations use. So in black, these are linear equations. In orange is a polynomial equation, and in green is a Bayesian approach. And one thing that all these equations have in common is that they assume that when you plug in the UK37 values into them, they derive annual temperatures, which might not always be the case because alkanones are produced by coccolithophores, and coccolithophores don't behave the same everywhere. They might have some bias in the data that these equations might not show. And a few examples of that, I have here a region in Brazil that's called Cape Frio, where this study found that samples close to the coast, they are cold bias, which means that once you plug in the UK37 values in equations, the resulting to temperatures lower than the mean annual average. And they suggest that this is a result from the upwelling system that happens in this region because of the westerly winds in the summer. Similarly, in the southernmost part of Brazil, right next to Uruguay, this study from last year also found a cold bias signal in their Alcanons data, and they suggest that this is because of the subtropical shelf front that moves from the La Plata River towards the Brazilian coast because of Brazil coastal current that brings these cold waters to the region, and that might cause their data to bias to colder temperatures. So with that in mind, during my master's, my advisors suggested that I tested these different equations in South Brazilian Bight, or SBB, to see if we find any kind of bias in our data or if they do reflect annual temperatures, and also to check which equation works the best in area. And why should we care? Why should we be testing these things? So here in a bigger picture, this is a benthic foraminifera record, but the idea is the same. So we as paleoceanographers want our proxies to work as want them to be as reliable as possible, to look in the past and see how climate changed through time. And that helps us to look what might happen in the future as climate might change or it is changing, but how helps us to assess what could happen as climate gets warmer. And this impacts local communities the most, because I hear an example of mullet fishing in SBB where it happens in the winter when mullet migrates from Uruguay towards Brazil. And it's when these communities raise a lot of their income, and it's really helpful for them to live throughout the year, and it's also really important for their food security. So understanding how climate might change this type of resource is really important for these communities. So here is the study area, the South Brazilian Bight. We collected 28 multicores on board of the Alpha Crucis research vessel from the University of São Paulo. So for those who don't know what a multicore is, here I have a few pictures. They are short cores, about 60 centimeters. And they're great for people or studies that want to look at more recent geological stuff because they preserve really well the sediments. And I use just the first centimeter of each multicore, so the core top. And because they preserve really well the sediments, you can use just the core top for this kind of studies. So the others, the samples you collected are the cross ones, and then the other symbols are just data that we're including in the study to increase our sample size. One thing I want to highlight in this image is the austral summer upwelling events. So austral summer in the southern hemisphere, December to March. These upwelling events are episodic events, and they tend to bring cold and nutrient-rich waters in the surface in the inner shelf. In the outer shelf, these waters reach up to 50 meters. Here is a summary of the methods. So I collected the K37 values from core top sediments. Then I converted these values into temperature using 6 different equations. I also used some climatological data from the World Ocean Atlas. So I got data from 0 down to 140 meters depth. Then I compared calculated temperatures against the observed data using some statistical methods. And here is a summary of the findings. So as you can see, the K37 values, they increase towards the slope, which means that in the inner shelf, the alkanones are they're registering lower temperatures than the outer shelf. And here's a box plot showing the distribution of temperatures using the 6 equations I tested. And as you can see, 2 of them derive pretty high estimates, whereas the other ones are between the same range, around 24 degrees Celsius. And then here is a big picture, I know, but I'm gonna go through it. Right there where you cannot see is each equation that I tested. So these are different equations. And then I tested different seasons. And here is from 0 to 25 meters in each column. And one thing I want to highlight is that in winter, it does not work very well. And values closer to 1, it means that the observed data compared to the calculated data are great, they work. And I want to highlight these 2 seasons, the fall, autumn, and summer, which are the warming seasons. And it happens to be when the upwelling system happens. So that means that our data is most likely warm bias rather than annual or cold bias like the other regions that I talked in the beginning of the talk. And we also can see that pretty much all equations work pretty well, but this two right here, the 2018 from turning singly, you cannot see it, but in the block parallel 2022, they work pretty well, and they also have the lowest residuals when you calculate between the observed and the calculated data. So if you didn't understand anything I said so far, just remember this slide. Can we use this proxy for paleotemperature reconstructions in Southwest Atlantic? Yes, we can. But we need to keep it in mind that they reflect warmer conditions. And that is most likely because of the upwelling system that happens during the warmer months, during the warmer seasons, that brings nutrients to the surface. And that makes the coccolithophores really happy. And as they're happy, they grow in abundance. And because there's a lot of them, they also die quite fast and they sink to the seafloor. And as they're living in these warm conditions, they skew the UK37 values towards warmer conditions, which when you apply the equations, you get warmer temperatures. And the next step to validate these findings would include doing sediment traps. So analyzing what's being exported down in water column and comparing to core top data, that would improve what we saw. And if you're curious to know more about the study and check my Q charts, you can look at my paper that was just published in Organic Geochemistry. And that's it.
It's a quick question about sediment sampling at the surface. As you know, in certain coring techniques, you lose a surface sediment. What's the best way that you come across to capture those upper sediments at the very surface?
So the multicore, it preserves really well, like it's pretty much intact. So when we are taking the water out of the multicore, you have to go slow so you don't disturb the sediment. So you push down and then the sediment stays intact and then you just slice it. Yeah. Yeah.
Divide that system variation.
Yeah, that's my second chapter. Which is from new for data, yeah.
So in the plot where you show just the surface samples and the temperature you get from them, were the two ones that were higher, were those like the polynomial, or is it different?
Yeah, polynomial and this one from in the 7. They result in really high estimates and they have the highest residual as well. So they don't really, I mean, kind of work, but if you want to be more accurate, we suggest to use these two equations. The last show that they're more recent equations. Yeah.
I don't know if you can ask this, but in one of your earlier slides, you say, like, if you want to use this proxy back to the Jurassic or something.
Eocene.
The Eocene,
Yeah.
you have to use species that are extinct.
Yeah.
How is that calibration?
Well, I never worked with them, so I don't know, but I know they exist. I know they're there. I've seen pictures. You can calculate CO2 with alkanones. That's how they calculate CO2 in the past using alkanones. I just never worked with them, so I don't know how it works. So, right.
Best part for the last before lunch.
All right, our last presenter for this first section is Beatriz Alejandra Aguilar, and she is a PhD student with Doctors Rosenheim and Romero, and will be talking to us about mangrove formation of below ground biomass.
Thank you. Hi, it's fun to be back to this class after getting core classes in here. Well, I'm gonna share some of my research. This is part of my chapter, and Well, it's gonna be kind of fun to present it. So this is a cute picture from one of our sites. We call it the fairy tales forest because it feels like you're in a fairy tale. So it's very pretty. We always use this picture. So as you know, I'm gonna talk a little bit about mangroves, which are really important for a lot of people interested in fisheries or people interested in like other animals because they're essential habitat, right? But they are also important for stabilizing shorelines and preventing erosion and protecting us from storms, right? Another function that they have as well is that they have the capacity to storage carbon. And we're very interested about that. And there's a whole concept about it. It's called blue carbon. So if you have heard that, it's very interesting. And mangroves are one of those that are very important in there. And why is this important? Well, they are helping us to mitigate climate change. We do believe in that. So then for that reason, mangroves have been impacted due to urban development. So there has been a lot of effort to restore them. But then one question that we have is, well, restoration is the regrowth or creating the conditions for mangrove to come here. So how do we measure that? How do we can make sure that the mangrove is being restored to its natural, to its original function? So another cool fact about the mangroves is that they have 10 times greater carbon removal rate than tropical forests. So very strong, but they also not a greater rate, but the capacity to storage even more. So they're pretty cool and they're pretty diverse, right? I tried to put this picture in here so we can see how this is the soil. This is how the soil look. So as scientists, we're very interested to understand a little bit more of what is happening above ground and below ground. So we, in this particular project, we want to ask how does the mangrove soil heterogeneity influence the carbon storage capacity? And in order to do that, we have 4 different sites that are allocated along South Florida, 2 sites in Tampa and 2 sites in Naples. In each site, we collected restore and natural as well. So each of our sites are different ages, and that's something interesting because we want to see how they vary depending on the time. So very important because not everybody speaks their language. So I'm gonna try to get us all on the same page. First of all, the below ground biomass. This refers to the organic matter beneath the ground surface, including live and dead roots, litter, and associated soil particles. The live and dead roots, so for this specific research, we classify it per size. So we're saying that everything that is 2 millimeters above, that looks like a root, we can classify it between live and dead. The bulk soil is everything that went down our 2 millimeter sieve, and that we classified it in 2 sizes. First is the coarse materials between 2 millimeters and 500 micrometers, and then we have the fine material. So that's from 500 to 45 micrometers. So important to mention for that blue carbon that I mentioned, we actually use the values from this part of like for the in general studies, but we are also interested to see what's happening with all the other components, right? So that's why I'm doing this research. Once again, maybe a little bit better shown in a map. We have our sites. This is the year since they were restored, so we have like a pretty good range from 30 to like 20 years old. And we have overall several questions. This is a big team, but specifically, we want to know how do vegetation, biomass, and soil carbon compare from natural to restore. We want to do this by quantifying the above ground carbon, peat depth, and in this presentation, I'm going to talk about the root dynamics and the different type of materials in the stock. So here is the methods. We go to the mangrove field, which is wonderful, very lovely. I do love it. We take it to the lab. We start a wet processing. We separate, we wash with the sieves, and then we obtain the dry weight. With this, we want to know what like is the proportion of the material. So basically playing with dirt, very fun. And then the extra step, because we were identifying there was a lot of different materials, is doing the loss elimination. So for those who received my email saying that's what I was doing, right? Technically, I was like, wow, how can we identify and separate these two types of material? And with that, and using the dry bulk density from our collaborators, which is a very important value of the soil, we translate this data to organic matter and inorganic matter. So basically, we can say now sizes and organic versus inorganic. We collected the core, we collected 3 replicates, and we used a Russian piston core, and in a 10 by 10 meters plot, we collected the different material, and we classified every 10 centimeters. So this is a 50 centimeters core. We did the processing. That was a lot of drying, a lot of mud. We classify by size, as you can see here. This is the classification that I was explaining before, but in a more visual way. And we obtained the dry weight. So with this dry weight, we obtained the percentage of material. And this is some of like the results that I already have. So in here, you can see the different type of materials, as I mentioned before. So we actually found shells in some of the sites. So we had to add this as another component of our proportions. And we have the coarse and fine material. So this is how our natural site looks. You cannot see there, but it's the natural. And we have our 4 sites, and they are organized among like the youngest to the oldest. Curious enough, they all look very similar. Basically, our fine material, it has the greater proportion in a percentage total, and all the materials are like very homogeneous. And then the question is, how does it look compared to natural? So to restore. So in here, we see that we are finding a little bit more of the shells, which is kind of expected because it's part of the restoration strategy. Also kind of expected for Schultz and E.G. Siemens to find this type of shells, and especially the sand layer, which was, the shells, yeah. And then if we compare like these two, natural and restored, they actually look very similar. So there's not a significant difference among them. Then I did some different comparisons. First, I wanted to see like if by interval there was any difference. It didn't look to me, we still can do like more statistic analysis, but in a visual way, like it seems like they're very homogeneous. And then we compare, because we obtained elevation, different elevation plots, we obtained that they are also very, very similar. So that's kind of interesting to find. Like I mentioned, we also find that there was like different composition and proportion, and that's why I put some pictures in here so you can see how like among the different sites, the replicates look very different, right? Like this was at least the restore side, and you can see like they do have like similarities, but some of them have like different depths. So it was something that we wanted to understand how different they were, right? So for that, we did this technique, the loss on ignition, which is a chemical test that we use to analyze the samples. Basically, it consists in heating a sample at a specific temperatures, and this will allow us to get the mass. And with that difference, we can calculate how much of organic and inorganic matter. So first of all, we remove the water, which is like a default. Then we obtain the organic matter with 550 Celsius, which is the one that produces a lot of smell. That's the organic matter part. And as you can see here, this is kind of like our coarse material, but you can see as well that there's like some organic matter in there. So some sites were very rich in that organic matter, and that was like interesting, and it's bringing like more questions for us, right? And then the last part is remove the inorganic matter to 900 Celsius. So like I mentioned before, we use the dry bulk density calculated and obtain the LOI, loss on ignition, and we have quantified this. So this is one of our data, very fresh and very interesting. So we can go from like our youngest site to our oldest site, right? So our youngest site, we found that the layer was very short, so they're still probably in development. We're talking about 20 years old, so it's very young. If we compare the ratio of inorganic versus organic, it's kind of what we expected, right? Because we have more sand, more shells, and all of that influence in here. This value is normalized to the mass that we weight, so the proportions of the material, the dry bulk density, and the section of centimeters. So that's why it's here. But just try to overall compare our data. If we see the organic, especially for like natural sites, it is kind of like expected. This is kind of like, it's kind of similar. It's like the same trend, but we are seeing some variation in this inorganic weight. And one of the questions that we were having is that blue carbon usually is defined as the organic carbon that is in the sites. But what if like this inorganic carbon also has like some potential? So that's something interesting. We just found a paper about that, and I plan to explore that path to see. The variability in the site is obvious, but especially in Henderson Creek, we found that our natural site, and this is supposed to be our reference sites, are showing interesting trends, especially being like the highest organic and inorganic material compared to, well, organic material compared to the other sites. So that's kind of like a first glance into the data. There's still some more comparisons to do and different approaches, but yeah. But just as take-home messages, the data compared by forest or by depth intervals doesn't show a significant difference. The presence of shells in restored sites is expected for the restoration strategy. Nevertheless, we saw that the older site didn't show that layer, so it's making us think that there's enough accumulation of other organic material in there, but something to still check. And the most abundant material was the finer fraction, which is very important in the isotope and blue carbon war, because if this is what is telling us how much carbon is there, then we are showing that that's the most abundant type of material in our sediment, so in the sediment core. So that is kind of like a cool insight. And this is preliminary data and might change when we transform this to carbon, but still working on the data. So with that, thank you so much. Questions?
Got time for 1 or 2 questions for sure.
I have a question about the restoration sites. How do you define that? What's that look like?
So basically they're planned by like different projects, like when you change like this ecosystem type for another reasons, and then you get like this law that says like, oh, you need to restore these sites, you basically go and you prepare like a whole study. The best strategy to do it is basically create the hydrology, like create the setting for the mangrove to come in there instead of planting. So like the mangrove kind of will like recruit itself in there. Some of the methodologies, I think, is like they plant like different type of like marshes, if I'm not wrong, and that kind of like helps and promote the mangrove to start like having the good conditions to come. So, and then it takes some time to build up the soil, and that's why we need to measure how is it evolving through time, you know. The densities, I will say it depends. Like I'm not presenting this data, but we do have like the above ground carbon, if you're like talking of like how the forest looks. And you can see the trend like within the age, like you can see more, and like we have already those values in carbon, so like you can see that the older the site it is, the more carbon they have. Oh, and but there's also like a latitude gradient that was like studied previously, so we know that the mangroves that are at the northern like Tampa sites, they have less carbon than the southern, because like they're more tropical, so they're more happy in the tropics.
That might suggest here, once you look into the carbon stable isotopes, might give you an idea of what your trends might look like.
Yeah, I think we are seeing a trend. It was like this project was trying to understand how like the different composition was among the sites, and if we find like differences, which we are, because we will expect like Windstar will have like, because it's the oldest, maybe like more organic matter, but then we have Henderson Creek just popping up like that, and we're like, but like I talked with my collaborator yesterday, he said like if things were like you were expected, they would not be as fun, right? So
Hey, everyone, we're gonna have a break now. Before you leave, remember to turn in your feedback forms, get a raffle ticket for this section of oral talks. Lunch will be in the MSL lounge, and we will reconvene here at 1:30. So that gives everyone a little over an hour. Everyone, thank you for coming back to the last. Thanks for coming back. This is the last oral presenter section of today's symposium. Just another reminder, we have physical copies of the feedback forms. And also don't forget to get your raffle tickets at the end of this section. So to go ahead and start, oh, and after this, we will, before we move to MSL for the poster session, we're gonna go do the raffle items. So don't leave before that. And then without further ado, our first oral speaker is Laika Confessor, who is a PhD student in Dr. Chappell's lab, and she's going to be telling us about seasonal trichodesmium clade patterns on the West Florida shelf.
Hello everyone, again, thank you for coming here. My name is Laika. I am part of Dr. Chappell's lab, and today I'll be presenting some preliminary data on chapter 2 of my dissertation, which is focused on seasonal trichodesmium clade patterns on the West Florida shelf. To get everyone on the same page, I want to talk about tricho a little bit. Trichodesmium is a type of marine cyanobacteria that's found at tropical and subtropical latitudes of the ocean. They are really important in ocean productivity due to the fact that they can relieve end limitation through their ability to fix nitrogen. And within the total estimated nitrogen fixation rate of 193 to 253 teragrams of nitrogen per year, trichodesmium contributes a significant bulk amount of that, so around 60 to 80 teragrams of nitrogen per year. And all of these end contributions can impact global nitrogen and carbon cycles. Within the Trichodesmium genus, there are 6 species, of which can be divided into 2 major clades. And for the purposes of my study and research, I'll be using T. tabadii or T. theib from clade 1, and T. erythraeum or T. ery for short in clade 3 as representative species of these 2 major clades. They have different optimal growth temperatures, iron stress responses, and growth alongside nitrogen fixation responses under rising carbon dioxide concentrations. And we know from prior research that T. theib is more abundant in open ocean regions, and nutrient limitation might be the driving force behind trichodesmium's overall nitrogen fixation rates and growth. So prior work confirms T. theib as an offshore clade. This is from chapter 1 of my dissertation. It's been published already. Woo! So areas focusing on the West Florida shelf, and it appears that low inorganic nutrient concentration is supporting these trichodesmium blooms. And there's a distinct biogeography around the 50-meter isobath. Here are some figures from my paper. It is showing relative T. tabadii gene abundances on the top and relative T. erythraeum gene abundances on the bottom, with that dashed line representing the 50-meter isobath. We chose this line arbitrarily due to the fact that within the coastal area of the 50-meter isobath, there is known underground wells and springs that might impact microbes differently compared to more offshore regions. So from these figures, it's separated by each cruise year for the columns. Specifically in the 2018 and 2019 cruises, T. theib is more abundant in offshore regions, while T. erythraeum is more abundant in inshore regions, where it's also positively correlated with coastal parameters such as dissolved iron and barium. However, One of our cruises was in June 2015, so different months, and there might be some seasonal things going on there. So this figure here is showing the absolute teeth erythrium gene abundances, and the squares represent data that was below detection limits, and triangles represent data that was below quantification limit. And here we notice that T. erythrium was actually more abundant in offshore regions. So what exactly is going on? This kind of leads me to the question I have for my chapter 2, which is, what are the seasonal patterns of the two trichodescent clades on the west Florida shelf? And I hypothesize that these two clades have different distributions, while in the winter dry season, there will be typical geospatial clade distributions observed, with T area more coastal and T theab more oceanic. However, during the summer wet season, I hypothesize that seasonal clade distributions might be impacted by the loop currents, where their intrusions might deliver river plume outflow to more offshore regions, where we might see an increase in T area abundance. So in terms of preliminary data, I have a entire pipeline created, but I tested it specifically on some data from the Sting project. This project has 2 cruises, one from the dry season and wet season. We sampled roughly the same stations in both of the cruises, with an increase in inshore sampling. This data is only about 50% of the total data that's available. I'm finishing processing those hopefully next week. So within this cruise, we coordinated sampling of trichodesmium, nutrients, groundwater signatures, organic matter composition, and isotopic signatures. And the parameters highlighted in red are what's available in our preliminary data analyses. And what's also interesting to note is that during the wet season in July, there appears to be a pulse from the Mississippi and Apalachicola river plume water that might have brought forth more lower salinity values offshore. And also that specific cruise was extremely hot. Evaporation might have occurred more inshore, thus increasing our salinity values in that area. In terms of methodology, we have a lot of data that was collected by our collaborators. So hydrographic data, that's our salinity, temperature, and fluorescence. That was from our ship system, so Seabird. Macronutrient and trace mail analyses were done in the Buck and Knapp labs, while groundwater signatures and radium isotopes were collected in the Tamborski lab. What I have done in the Chapel lab was analyze and collect chlorophyll A samples, as well as look at DNA RNA sample collection, extraction, and quantitative polymerase chain reaction, or qPCR. Very briefly on the methodology, basically we got water from each station via towfish or CTD. These are all surface stations that I'm looking at, and we filtered them onto Sterivex filters. Next, in the lab, I extracted them using an all-prep DNA and RNA mini kit. Or for the DNA, I used the Rocco et al. Protocol to quantify each clade gene abundances with the RNPB gene. The RNA I'll be using for future work in my chapter 3. So yes. Statistical analyses, there's a lot. I'll be going over part of it briefly. So with the combined data set, we have our species abundance data that we collected via qPCR. I did a log-tan transformation on that. And then for our environmental data, so silicate, chlorophyll, phaeophyton, temperature, salinity, and bond depth, I did a z-score standardization on that. The final environmental data is still a work in progress. I will probably include more as we get more data that's available. So with that combined data set, I did a bunch of exploratory data analyses. I also categorized variables based on inshore and offshore variables, as well as our seasonality patterns. So for the full data set, so all of our combined data with no seasonality separation, I did a cat plot, permadis, and permanobust, specifically looking at the isobath, just trying to check to make sure that the 50-meter isobath is still a good indicator for offshore versus inshore samples. I won't go in depth into that because it's a lot, but just know that the 50-meter isobath still holds true for like analyses. Next, I looked at each cruise separately, and then each season separately. But the one thing I'll be focusing on for the rest of this presentation is DBRDA with AIC. And what exactly is that? Good thing you asked. DBRDA is a distance-based redundancy analyses. It's basically a constrained type of canonical analyses using Bray-Curtis distances. We're trying to look at how much variability in our response species variables is explained by predictor environmental variables. I took it a step further and included a stepwise selection with Eckheig information criterion. I hope I pronounced that right. This is a stepwise process to just see which group of our environmental predictor variables affects responses the most. And Typically, the group with the lowest AIC is the best model that describes our responses that we see observed. That means that not all the environmental parameters that are included in the model will be included. Yeah, so moving on to our DB-RDA plots, we'll be seeing a lot more of those, so I'll go into one each in more depth. So the stations you see here in black are our sites specifically. The closer they are to one another means they're more related or they're more similar. Our red is our species data, so our T. Erie and T. Theab. And our blue is our vector environmental gradients that were more significant. So these are all the environmental parameters I included in the model. The ones shown are only the ones that were significant. And the x-axis holds the most variance explained, around 36.31. This is including all of our 2 combined sting cruises. And what we notice is that T. Theab is mostly explained by increases in bottom depth, while T. Erie is explained by increases in temperature and chlorophyll. So these are typical trends that we expect to see. However, that variability explained isn't good, so I decided to take a step further and separate the 2 cruises separately. And they're also in different seasons too, so it made sense. So moving on to the dry season in February, off the bat, we noticed that the Kaggle axis 1 explains a lot more variability, around 70%, which is pretty good. T. Theab is associated with increases in bottom depth and salinity. Oh, I also forgot to mention, for this specific cruise, I was also able to include K. brevis species parameter because we were able to get cell counts. I probably won't include this into the rest of my analyses, just specifically for this cruise. T. Erie was also more associated with increases in 224Ra. This is an isotope that tracks submarine groundwater discharge, so more of our coastal parameters, as well as chlorophyll and silicate. So again, we're following more typical coastal offshore parameter trends. It's a pretty beautiful graph. It makes sense. And then we went to the wet season. Things get a little bit weird here. So the variability explained is a lot less, and there appears to be actually reverse salinity and that 224Ra trend. So decreases in salinity was actually associated with increases in T. Theab, which is not what we expected. And T. Erie was associated with decreases in 224Ra. So not what the typical geospatial trends that we expect to see in our area. But again, this season in particular was extremely weird. We had that river plume that was being carried by loop currents, carrying more of that low salinity values offshore. And that general cruise was extremely hot, so evaporation occurring at more coastal areas of the shelf. So yes. The basic gist of this, so the main takeaway, is that environmental parameters best explaining trichodesmic clade variability differs between each of the seasons. So we see general trends in both the full model and the winter dry season models following the observations from the chapter 1, where T. erythrium is more coastal and T. Theab is more oceanic. And that it appears that the winter dry season is better explained by environmental data. However, loop current intrusions and temperature during the summer wet season impacted trends that we observed in that particular season. And T. Erie was observed at more offshore stations in summer wet season compared to the winter dry, as you can see over here. So what's next? Basically, I had to finish qPCR and include the rest of our data. I will complete analyses of the combined data set. So there are around 5 other data sets I'll be including in this chapter. And then that will also include finishing our DB-RDA plots for that, as well as including any additional data from collaborators when they are available. So yeah, I want to take the time to acknowledge all of our collaborators in the STING project, as well as the Chapel Lab who have all contributed to this research. So I'm happy to take any questions now. Yes.
It makes sense. You divide analyze your data in terms of dry and wet season.
Yeah.
It's 50 meter as best. I don't really know about that. That's going to be about some categorization of seasonal changes. Also, you mentioned that the loop current intrusion. What does that mean? Intrusion to the 50 meter as best?
No, hang on, let me pull up that slide. Basically, the loop current intrusion is more affecting our offshore areas. So like.
You mean intrusion to the the West Russia?
Yes. So like, that's what I'm hypothesized. Yes, sampling area.
No.
The western sample.
The western.
Yeah, so right here.
See the current, like, loop through, grab the river and pulled it into our sample.
Yes,
But it didn't do that.
when you talk about the Western Shelf and intrusion, enclosures, intrusion really means, intrusion on the Western Shelf, that means intrusion. In the Gulf Vessel eastern part, it's just a kind of variation or evolution, pattern that's fine. Intrusion, the word used in the context of Western Shelf, that means Western Shelf intrude onto the Western Shelf.
Gotcha. Okay.
That's
To a specific
that means those two different processes.
Okay.
Oh,
To a
okay.
specific isotope to get it to be officially called.
Intrusion usually occurred on the outer Western Shelf, for example, the shelf break 200 meter as best, and that it is offshore from 150 meter as best. That part area we you can see local intrusion. And it's further onto the shelf. Usually local cannot get that close or to shallow water on the Western Shelf. So when you generally talk about local intrusion, you don't want to show this SOS. I'm really concerned because we talk about the observation mostly on the Western Shelf and the intrusion. So we'll be vocabulary. Thank you.
Yeah, thank you so much. Yeah. Yes.
I have a question regarding geology, how it might impact your study. That Florida platforms like Swiss cheese.
Yep.
The carbonate dissolution occurs during the glacial periods, and so all these caves and places where potentially low salinity waters may come up, and like in your Fort Myers, Mudhole Submarine Springs, and there may be other places off the West Shelf. I was just curious, could those potentially impact your study in terms of new kind of water masses coming up where you might be sampling? I think you're further north from Fort Myers, but I'm just curious if that might be something to consider if you do broader sampling up and down the shelf.
100%. It's definitely a factor that we are considering. I know we sampled some blue holes. I think it's like the green banana blue hole it was called. Yeah. We do have some samples for that. I'm still in the process of like getting that data like extracted. So I'm hoping that we can include that here so we can see those trends. But yeah, definitely I think there could be some effects on like the trickle distributions and stuff.
Warmer and lower salinity.
Yes, yes. Thank you.
All the questions we have time.
And I forgot to mention, we refreshed all the copy and stuff, so feel free to get some more. This is yours. No, this is. Should be. This one? Yeah. All right, our next oral presenter is Majid Hajjal, who is a postdoctoral researcher with Doctors Hugh and Barnes, and will be talking to us about those floating materials in the Northern Adriatic Sea, the observations from satellites.
Hi everyone, I'm Majid. Today I present you a study about the Northern Adriatic Sea, floating materials we've observed from satellites. The Northern Adriatic Sea, it's in Europe, the east side of Italy. Here I'm showing you a satellite imagery of last summer, August the 2nd. For comparison, this area is approximately 20 times the size of Tampa Bay. So for comparison. And on the left side, I'm showing you an RGB image. It's what you would see if you would be flying next to the satellite with your eyes. On the right side, I'm showing you a false RGB image. We just replaced the green color by infrared signal because the vegetation has a very strong signal in the infrared, but not the water. So it's very easy using this technique to see floating materials in the ocean. On this image, we see different things. So we have some clouds in white. We have the land, and in the middle, we have some greenish colors, likely phytoplankton bloom, not sure. And in the middle of it, it's much better to see it on the right image, the FRGB. Some white, yellow features, surface features. they are quite large. You can see the scale here is 20 kilometers, so it's over 100 kilometers of material present at the surface of the sea during last summer. So we had to investigate what was it. When you don't know something, the first step you do is literature review, obviously. And from the literature, we have three main candidates. The first one is marine mucilage on the left. It's polysaccharide, it's organic material produced by some phytoplankton species, also from land, from seed and roots. So it's not only for marine biology. In the middle, we have red Noctiluca sentiens, which is a big dinoflagellate. You may have experienced it as sea sparkle because it bioluminesces at night, sometimes on the beach. And the last target possible is plastic. The recent study from last May, they proposed that this exact area, it's a hotspot for floating plastic. The red, it means a lot of plastic, so likely in the west and north part of the area. So how do we analyze that? We need to process data from a remote sensing perspective. So I processed the last 45 years of satellite imagery over the area, which represent approximately 15,000 images. I take all these images, all this data, and I make it in RGB and FRGB, as I've shown you before. And when I see something on the FRGB, as you can see on the bottom left, I cycle it, highlight it in red to say there is floating material there. Then once I've done visualization of everything, I need to identify the target. What is it? And for that, we have various material. The most common one in optical oceanography is the spectra, the light spectra. So they look different. We also have in situ data reports from different agencies. So it's useful to do that. And once we have identified the different materials, we can make composite, morphic composite and your maps. So the first material I said is mucilage. Here you can see on the right, it's a picture from Istanbul, so in Turkey, Eastern Europe. Summer 21, it covered the whole area. Here it's a boat, and it's quite a large boat. So someone put his hand in mucilage. The nickname of mucilage is sea snot. You can understand why. And the main supposedly producer of mucilage is dinoflagellate. It's called Coniolas fragilis. It's a picture on the scanning electron micrograph on the left. And it's supposedly outperform other species. So more species produce mucilage, but this one is up to 5 orders of magnitude more efficient at producing it. And one of the suspected reasons for, at least for seed and roots for mucilage production, is to protect from the sun. But we don't really know yet for the reason, the mechanism for in-water marines. Here it's a picture from MODIS, so low resolution on the left, high resolution on the top right. I try to cycle around where I can see mucilage. It was in 2004. It's just an example. And on the bottom right, it's what you would see from a ship. So it makes this kind of creamy layer. And mucilage itself, the danger is it can clog and suffocate benthos and pelagic fish, so they cannot breathe. So that's the main issue, the main environmental issue, along with tourism and fisheries. The second target, it's red Noctiluca sentient. Here I'm showing you a little video of it. It's the largest, I expect, dinoflagellate. It can be up to a millimeter in size, so it's quite big. And here it's feeding, so it's basically a big balloon with a tentacle. And it's trying to catch anything that crosses in front of it. It's not toxic. That's quite important. It makes red tide, but doesn't have a toxin that is harmful. On the right is some light micrograph pictures. So it's A, B, and F, it's detritus. So probably just ate something and it digested it. C and D, it's diatoms. So it's very good, very efficient at hunting diatoms. And E, it's even more interesting, it's copepod eggs. So it's phytoplankton that can eat its own predators. So I find it quite interesting. This is what it looks like from a remote sensing perspective. Again, on the left, I try to highlight around where I see Noctiluca in 2017. Here on the top right, it's high resolution imagery. So it's orange. You make this orange color, as you can see from a ship observed here, and these long slicks orange at the surface. I said it's not toxic, but it can still be harmful. So it's considered a harmful algae because it can cause, of course, hypoxia when it dies. And when it dies, it also releases high ammonium concentrations, which can kill fish. It's harmful to fish. It happened in Florida in 2011 in Pensacola. We had February outbreaks of dinoflagellate, of red Noctiluca centella, which killed fishes. The last target we observed, so it's even more east of this area. It's in Croatia waters. We had this white, yellow surface features aggregation we observed. It was in May 22, so that's the same day, but different hours, and this is high resolution. And we were wondering, was it plastic? Because that's what of the report, and we didn't see it yet. Here, the size is 10 kilometers, probably. It turned out it was not plastic, but pollen from pine trees. That's our main hypothesis, because a recent study from Chunmin and Al from 2 years ago in the Baltic Sea, so the Northern Europe, Scandinavia, they observed pine tree from pollen from pine trees, so it's yellowish, and it makes this kind of slicks at the surface of the ocean. So it's very similar to what we observed in this area. It's at the same time in May, so it's coincided with pollen release, and it's not been reported in the area yet. So once we have identified all of these projects, we can aggregate them into temporal maps to see where they are distributed. Here quickly, I'm showing you where mucilage can be found. So you can see on the bottom, it's mostly in summer, June to September. And the extension varies. 2024 so far is one of the worst years in terms of spatial extension. It's almost the whole area, but it's not continuous. It's not a single day. It's 3 months. It's over 3 months. It's just spread to various areas over the months. I can do the same for Noctiluca. This one is in spring, so February, or late winter to June. Similar distributions, maybe sometimes it's very small. 2021 was the last outbreak in the area, and it was also quite large, almost the entire area was covered. And the last one, pollen, so it's on the east part of this area. 2022 is interesting because it expanded in the center of the Northern Adriatic Sea, also in the north. So maybe it's becoming an increasing problem. Now that I've shown you the spatial distribution, I'm showing you the temporal distribution. So here on the x-axis, it's the months of the year. So on the left is January, on the right is December. And on the y-axis, it's the years. So 1980 is the first outbreak we cannot see because of this little thing. But there is a Noctiluca outbreak 1980 is the first one observed. In blue, it's mucilage. In red, it's Noctiluca, and in green, it's pollen. So what we see for mucilage, it was quite intense from the 1998 to 2004. We had many outbreaks every summer almost. And then it stopped until 2018 and 2024 last summer. So it's interesting that people were predicting that it's going to get worse because of climate change and these kind of things. It's the opposite for Noctiluca. We had only 3 until 1998 as observed from satellites. So every 9 years we had an outbreak. And since 1998, we had like 12. So it's an outbreak every 2 years almost. Of course, they cover different surface. And some pollen that is likely increasing, the presence is increasing in the area lately. But it was not reported before. I can aggregate both the maps, temporal maps, and the spatial maps and temporal distributions into to have an annual presence. So the chance to find one of the material at an area every year. So you can see mucilage where it's distributed. It's mostly on the west shelf of this area. And what I didn't tell you, there is a river, one of the major rivers of the Mediterranean Sea there. So it's just around the delta of this river. Mucilage is distributed. Noctiluca is different. It's 1,000 more south of this area. They both cover the entire Adriatic Sea, Northern Adriatic Sea. Pollen is concentrated to the right side. At the beginning of this talk, I've shown you a picture from another paper, recent paper about plastic. This is what they report. So hotspot is likely the west and the north part. So the left, the west side, it could be mucilage for this, not plastic. Actually, we didn't really see plastic. Some reference for the people online that can pause on the video. Take a message. So we didn't really see plastic. Actually, it's hard, even not possible currently with the current sensors from space because the concentrations are too low. So a single plastic bottle, you cannot see it. And microplastic, they don't really alter the light signal much yet. Satellite data, they agree with in situ reports of the various material present, except pollen because there is a lack of in situ studies. Noctiluca seems to increase since 98, every 225 years, an event. We didn't have for 3 years, so maybe 25 is very likely. Don't know if we can make some statistics, but very likely there is an outbreak this year. No particular mucilage intensification in terms of occurrence. 2024 was quite large. It was one of the largest, so maybe there is some. And it's the first time we report pollen in this area from space. Thank you. Yeah, thank you.
Question about the pollen. Are the pine trees located in certain areas that correlate with where you find the pollen in the water?
Yeah. Actually, there is just one single study in this area. It's actually, there is a city split. I don't know if you know, it's Croatian city there, and they have airborne pollen in this area. They don't have it here, so they report it there, which is 200 kilometers south. And pine trees is the main producer at that time of the year, from February to May. Is the number one. With another grass, forgot the name.
I think the surface current, maybe bringing it up there.
No, it's the air that is depositing it. I don't know. I don't remember if there is a time until it disappears, but it makes this blurry effect. It's just sinking in the ocean. So mucilage, it's kind of stopped in 2004. We are just since last 20 years. It's the opposite for Noctiluca, the red. We had like one every 9 years for last two decades. And the last 2 decades, it's 10. So I don't really know. This agrees with in situ observation. So I don't know for mucilage, they were all predicting it's going to get worse because obviously when you start to see that it's becoming a big problem, it's like if you see red tides every year, you are predicting it's going to worsen. It didn't happen. For mucilage, for Noctiluca, I don't know. I think maybe it's a global thing. We are seeing more and more. Noctiluca is very efficient at feeding on diatoms. It's a winter spring thing. So maybe if there is more diatoms, more food they can develop. So maybe there is a hidden link somewhere, but I didn't see.
Data for those different kinds of samples and do for that.
For sure. But also feed on copy pods, so I'm not entirely sure the real link. Yeah, but yeah, that's a good point.
Okay, I think that's all the questions we got. All right, our next presenter is Kiyu Mao, who is a PhD student with Dr. Who, and is going to be talking to us about tracking brine shrimp cysts from space. Take it away.
Hi everyone, my name is Ke Yumao from Optical Oceanographic Lab. Today I'm going to show my research about tracking brine seas from space. That's a case study of the West Aral Sea. First, I'm going to introduce the background. So first, what is brine seas, BSC? They are tiny eggs produced by brine shrimp. These eggs are very special because they can survive in extremely salty water, such as salt lakes like the Great Salt Lake. And second, they are served as superfood for fish. That is very good for aquaculture and value of great value. That's tens of millions per year. And so how do we detect them from space? That's a global map showing the distribution of this brine shrimp. There are two aspects that we can distinguish them from other species from the space. First is its special shape or BSA. BSA patterns always called aggregate. They are tiny and floating on the surface of lake, so they can aggregate into a large patterns called slits. These slits are very large. They are up to several kilometers in length and hundreds of meters in width. So it's like long strips. And second is from its spectral shape. That's Majid also said that the spectral shape is kind of like a fingerprint of each items. Different elements can reflect lights at different wavelengths. So for the special brine shrimp cysts, they have wavelengths at inflection points at 550 nanometer and 650 nanometers, shown in this figure. And this figure also shows a great match between the lab measurement that's more consist, and the black is our reflectance from the satellites, because satellites has only several bands, so it's look like straight lines. And this spectral shape also increased from 550 nanometers to 800 nanometers. That's our basement to detect BSC from the space. So by knowing the key points to detecting brine shrimp cysts, I'm going to introduce my study area, the West Aral Sea. This place tells an amazing story of change, because in the 1980s, you can look at this figure. It has a boundary of, in the black line, that's a very huge area called the Great Aral Sea, called Aral Sea. And after several years, it has doing transformation from a freshwater baseline that has a salty from 10 grams to very salty water body that's up to 180 grams per liter. This change has caused the transformation of species in this area. This once abundance in fish has changed to brine shrimp, and this brine shrimp has taken over 99% of biomass in 1990s. So this has a perfect place for our study to look at it as a natural laboratory in our study. So in this study, we want to ask 3 questions for this. So first is, where are BSC in the West Aral Sea? And second is, how do they change over time? And third is, what drives or causes these changes? So for the satellite part, we use Sentinel-3 Ultra data from 2016 to 2023. We use this satellite because it can view the entire lake in every 2 or 3 days. That gives us very frequent updates on these BSA changes. For the flow chart, you can see that we have 3 main steps of this. The first step is to clean the satellite data, like removing the sea clouds and do some correcting for atmospheric effects. And that gives us clear images to see the brine shrimp cysts in this lake. And second part is to build deep learning models. That is, try to use deep learning models to automatically extract BSC in these images. Think of it like teaching a computer to look at the brine shrimp cyst patterns, like what our human being eyes can see these patterns. Finally, after using this model to extract the results, we can calculate exactly how much BSC
and is present and create time series maps to show the changes. So let me show you this, what's the model from. The first is show the works with a real example that in October 2019. These three images show the same view, but in the different ways. The left images, that is false RGB images, that is to looks better of the BSA slits. This cannot look very clearly from this screen, but you can see on the second images that the BSA slits are highlighted in the NDVI, that's normalized vegetation index, that can show the vegetation more clearly. And third, it shows the extracted results overlaid by the BSA slits. That's scale bar, color bar shows the density of the BSC, ranging from the blue to the red. Red means there is a high density of branching cysts. So this case shows that our model can pick up BSC from other features in the lake. So now let's show you how we learn when we apply this model to 7 years of data. Looking at 7 years of data, we can see clearly patterns in where and when we find the branching cysts in this lake. First, look at this monthly maps on the left. The color shows BSC concentrations from blue to the red. That means lower to high. You can see clearly two patterns here. That is, one pattern is in the September and October, there's a peak season in the BSC in these regions. And the second pattern is that the most BSC slits are always occurs in the southern and western part of this lake. That is the special pattern. So how do we explain the spatial distribution of this BSC? I look at the wind map, the wind data. Here is the Rose diagram of wind speed and directions of different seasons. You can see that the predominant wind here is from the northern and eastern here. So that blows the BSC from the center to the western and southern of these lakes. This explains why we see consistently find more BSC in these regions. So now we look at the time series changes of BSC. These three maps shows three different stories. The top graph shows monthly changes in branching cysts. You can see how BSC jumps up and down through each year. But when we average this into a typical year pattern in the bottom left, we can see the similar pattern that it has peak season in September and October. And the most interesting part is on the bottom right, from the yearly change in this region. The yellow bar shows our model findings, and the red line shows the data from BSC harvest from local government. You can see that they match clearly. You can see that it has three trends, the three periods. That's from 2016 and 2020. It has gradually increased in that area. And there's a sharp decline in 2021, and then gradually recovered afterwards. So we have two assumptions for the sharp decline. The first is there may be a cold, there's a cold spring in that season. That may cause the development of branch shrimp that's a little bit later in that year. So may cause not that abundance in that year. And the second assumption is that in 2020, the human harvesting activities are very huge. So that may disturb the whole community of branch shrimp. That may cause the decline in the second year. So what caused BSCs in these patterns? Our analysis revealed two key environmental controls. The first is food availability. Here we use chlorophyll A as the index for the food availability because branch shrimp is phytoplanktons. So we can see that the chlorophyll peaks in the April and May and lower in the autumn. That makes sense that why BSC harvests in the autumn season, because when the food scarcity in fall may promote the branching seeds to produce seeds for their survive strategy. And the second is the bottom is the temperature. The water temperature shows that it's increased in the spring and higher in summer and lower in the autumn and fall. So that's explain why we see the branching seeds harvest in autumn, because branching seeds can hatching when the temperature over 5 degrees C, and when the temperature is too high in the summer, it will surprise the reproduction process of the branching. So then, when the autumn, when the temperature is very good, that for maybe 20 degrees C for the reproduction, and the food scarcity happens again, that causes the peak in the autumn area. And look at the right graph of this. That shows the different environment factors change yearly. You can see the orange dashed line here. That means March and April average temperature. You can see that in the 2021, there's an extremely decline to about 2 degrees C in that year. And for the most of days is often below zero of that temperature. That may cause the delay of that year's hatching average. So these findings has important implications for BSC management. By monitoring these environment factors, we can better predict and perhaps even optimize BSC production. Here's my key takeaways. Deep learning and spectral mixing enable remote detection and quantification of BSC in the West LC. The second is wind drives BSC spectral distribution, and most BSC is found in the west and southwest of West LC. Third is BSC shows strong seasonality, with September and October being the peak months, which are linked to the temperature and food availability. Inter-annual change appear to be driven by temperature salinity and food availability, but it needs more evidence to prove that. Thank you. Yes.
I have a question about the salinity change. In the beginning of the talk, you mentioned it was around 10 and went up to 180 or so.
Yes.
What caused that big salinity change before the BSC arrived?
Because there's two river inflows into the West LC region. And during the 1980s to the 1990s, there's a huge increase in the agricultural use of these two rivers water. And there's almost no inflow of one of this river, and another river has seen an increasingly decrease of the river water input in that area.
BSC was already there, but it just started.
No, BSC is bring from other places, like birds. I can bring. Yeah. Yeah, they can sit and they can stay on the lake on a flow on the surface or at bottom for a couple of months or a couple of years, because it can, when the temperature or the salinity is good for its hatching, it will hatch into branching. Oh no, it has two strategy. One is to become live birds. That live birds, when the temperature is higher, you see that the BSC abundance is very low from the March to August. That's because the live bird strategy dominates this process. And when during the winter, it has to survive in the cold winter, so it can't to reproduce this. Yeah.
Are these same?
There are different species in the globe. There maybe five or six species globally, but in the general, their spectral shape are similar. Yeah. Yes.
You saw in some of your abundance maps there was like a February peak.
Yes.
Do you have any idea what that's called by?
I'm also very curious about it. So I have think about it, and I have maybe one or two assumptions.
because one assumption is that during the winter, the wind speed is a little bit higher, and the observation days will be lower because of more cloudy days. So that may cause the data to February data have, yes.
That's all we have time for now. All right, and then last but certainly not least, we have Seven John, who is a postdoctoral researcher with Dr. Liu, and who will be talking to us about a coupled physical biological model of Karenia brevis on the West Florida shelf.
Hi everyone, this is Sepin working with Yongang's lab. So today I'm presenting the topic on coupled physical biological model of Karenia brevis on the West Florida shelf. So this is kind of an application study for the red tide in 2018. So before I'm going, I will just brief my outline structure. First, I will explain about the circulation patterns. Then I will go briefly idea about how that happened in the 2018 red tide. Then about our modeling part. So already you had two great talks about FECOM, WFCOM. So I just explain what that little bit and then go to the biological model procedure. Then we go for the simulation and corresponding results with that. So the red tide. So in Florida red tide, it's a complicated process, like complex process, perspective of the biological process and as well as the ocean circulation patterns. So the first figure which I showing is a spatial distribution from the period of 1953 to 2019. So you can see how the Florida red tide is happen or Karenia brevis samples distribution is there. So the major distribution is happened between the Tampa Bay to Charlotte Harbor estuary. So that's in between epicenter, and there is a one circle shape shown. That's maybe the formative region for the transport based on the circulation pattern. These are the previous studies which I taken from the Weisberg 2019. Another one is circulation. So circulation, there we have a pooling circulation. So basically means a pooling circulation. There is also another loop current interaction. There is a pressure point. So if that pressure point interact too much, then upwell favored condition will be there. If the species is there, the upwell favoring more to be generate or transport towards to the coast. These are the previous studies. So now I go to the 2018 red tide happens. So that's from the January to February, but I put the name title as 2017 to 2019. Why? Because when you see the January onwards, the cell concentration already started. So that means it is previous year that started the blooming and it's continued to the 2018. Also, it is continued to the next year up to the February. So that's why I put the title as 2017 to 19, but we are representing 2018. And the black color is representing the central and southwest Florida, and the red indicating the northwest Florida, and blue is indicating the East Florida coast contribution. So you can see that when East Florida started in September after that, and it is expanded in October number. Please remember that point. But come to the WFCOM. So already you heard about that. What is that? I want to express only that last 10 years is working with West Florida coast solution model. We have plenty of applications. Most recent application about the red tide is 2019 and 2022 and 23. So one is 2000 red tide in hypothesis, how that happens. And 2022, the termination process based on the physical model. And 23 short-term forecast for the trajectory for the Karenia brevis red tide forecast. These are the recent application from the our WFCOM model. And this is the domain figure. That's just only for express that we have a high resolution, better capture for the spatial features. Now comes to the 2018, a lot of hypotheses there. So we follow my work is followed by our work is followed by the Weisberg
2019 work. So they deployed a glider. So from there, they get an information about the different parameter, but I have put it only chlorophyll, which our industry is chlorophyll. So you can see that some dark red on the, I put it in a round circle. So those parts are interested, and the glider points, which I marked in the second figure with red cross, you can see the different red cross points. So those are the path followed by the red glider data set. So in subsurface, this chlorophyll, high amount of chlorophyll found, then further investigated with the lab, then they confirm that these are from the Carina brevis cells. So after that, WFCOM particle tracking, here only physical model, but based on the circulation from these points, is there any chances to come to transport to the coastal area? So that is represented here, that figure, the line pattern. And the best interesting thing is those particles are in the subsurface area, and the depth color is varying from blue to red. So the surface is red. So when it is reaching near to the coast, it's surface area, surface layer. So those circulations basically by the upwelling loop current interaction happen in the pressure point. So all the theory was presented only along with the physics. So people ask that, okay, how the biology process can be incorporated in this work. So we come up with a coupled biological model. That is our generalized ecosystem model. That is epicome based model and the ecosystem generalized. That means we have a flexibility to incorporate the different species and taxa and the model. So we limited with minimum configuration to understand the interaction between the different species and different environmental conditions. So for that, we used 12 functional groups. All the functions are I listed in the second figure. So basically NPZ model, I just simply plotted in the first figure. So this is capturing the physical information, light information, salinity, temperature from the WFCOM. Then we do the ecological modeling patterns. So in that, we put a one species as Carina brevis, and also we incorporate diatom. But we are currently dealing only the response of the Carina brevis species. So a lot of parameterization are incorporated in the model. I just demonstrated this. These are not taken from our, we taken from the previous literature. Some of them are from the epicome application studies, some of them from the Florida Carina brevis studies. Now comes to the procedure. So whenever do the modeling, we need a good initial condition for get a good results. So we took a hypothesis from the glider data set. Those are the points marked in the particle tracking. Those regions we considered as a high chlorophyll amount. So we initialized the model with the subsurface layer with low concentration cells. So that is low concentration of the only in the surface, subsurface, not in the surface. For the convenient purpose only, I plotted two cells as shown in figure. So the subsurface cells, that means the model we initialized only in the subsurface layer, and the other color shade, which I show in, that is observed location from the FW datasets, we put it in the surface of the model. And the color code, which is represented similar to the FWC and showing that very low is white color and low is yellow, and corresponding red is the high concentration. So we concepted with the concept of that existing cell is there that's previously come from the 2017 onwards to the system, and also we newly initiated cells at the subsurface layer. We conducted a 60-day simulations. So these are the comparison with the observed data from the FWC and the model comparison. So the first first figure showing model, and the second panel is observation. The dotted points are the observation points. So that's this comparison is showing after the 10 days after the initiation. So that is, you can see that matching with the surface with the color code patterns, and also there is a coming from the surface from the where the surface layer transport. So after 15 days, again it comes to the 25. That's in the September 25. you can see that's a shift from the previously. That's advocating also contribution from the offshore initiation cells to the coastal area. This is after the 25 days. Now comes to the next month. That means October. So that's further expanded and transported towards the Florida Keys. So the advection based on the circulation patterns, you can see that. So the Florida Key here also, we are getting high concentrations. So that's the pattern is matching with that. After that, we compare with the satellite data that's collected from our collaborators whose labs. So we compare with that. First one is showing model, and the second one is the satellite derived product. That's not a satellite product, satellite data. I just reprocessed where the cells, binary cells having concentration more than 100,000 cells. So that color is showing here. So those patterns are as matching and correspondingly with the observation also. So here the criteria is given 100,000. Sometimes they have more than 100,000 also. That also in our model is getting there. So this is the what I said previously. So this is the simulation. What I was showing how the transport is happening for the next 60 days and how the adverting and the concentration, the patches is changing from the September to October, and the distribution of the concentration cells also changing. So we can come up with a summary that we implemented a coupled physical biological model for the Carina brevis for the West Florida shelf and used 2018 red tide with the both offshore source of new cells and existing coastal patches from the cell. Both in situ and satellite imagery are used to do model veracity test and demonstrated the importance of the coastal upwelling circulation and organism process of the biological process. And anyway, we know that we need a lot of improvements in that fine tuning is required. So now, thanks, almost I just showed in a study. So you can see that how the coastal, this is the surface layer, this is the coastal patch. So then initiated in the bottom, that's the bottom profile. So that how that's coming and integrating and the cell concentration changing over the time. Thank you. Yes.
So the origin is on the seafloor. Is it kind of protected from impacts from hurricanes in the sense that the hurricanes may push water around on the surface, but leave the source in the sense?
Yeah, it depends upon the where the cells are there. If it is too bottom or how the effect impacted on how much bottom. If it is bottom happens, then it will be
Display.
display.
Otherwise, it
Yeah,
still
yeah,
continue
yeah.
on in the storage.
Yeah. Yeah.
Question that's NPC model, right? So,
Yeah.
There's terms that are consuming. I'm assuming there's some sort of consumption of cells.
Yeah, actually that basically we tested that we didn't incorporate it here. So that currently we just give the two zooplankton configuration is there. So based on the growth rate and decay process, so that we simulated that part also. So, but in this here we showing only the development.
Because it looks like it's just like spreading, but in like real life and in an NPC model.
Yeah.
NPCD model,
Yeah, it is degrading. It is there. But this time we just only with the development process. So if you run with the 60 up to the next January onwards, we have the results. We didn't incorporate here. So the termination is there. So gradual over the time, disintegration is there. Not pretty much, but we are getting that kind of result because before doing the production, we just tested that kind of process also.
Nutrient inputs solely heterogeneous comes from river inputs or is there
I forgot to tell that. Sorry, thank you for remembering that. Actually, currently we only initialized in the water column, not from the external source. We didn't incorporate it now.
So there is no river inputs. We have the river inputs, but nutrient information is not incorporated in this simulation. Next step is to be incorporated grab the data information for the like ammonia, nitrate, and those kind of things. Right now, we initialized with the Atlas, global Atlas data.
Well, my interest is not just what's coming off the land, but what's also being upwelled.
Yeah.
And the balance between those.
Yeah, yes.
Is that something you could look at?
Yeah, right now we didn't incorporate it. We can check out that. Yeah, we can do that later stage because once stabilize the model, then we can respond.
One more quick question. Mine is not fit, so someone.
Okay, no more questions. All right.
I'm actually impressed by the model result. It shows very nice agreement. With the in situ sample with the satellite image. The question is, what environmental variables are most critical to lead the success?
Yeah.
Or if I put it the other way, what type of information can you not afford losing?
Yeah.
We all know how complex.
Yeah, it's because if you ask the why red games, the process, it's very complicated.
Yeah.
So what we did, a lot of assumptions we considered. Based on that we take it some point of information. One is if the model is not getting proper upwelling feature, this kind of feature will not be create like that adversion transport that will not come. Another one he thinks we need a good like information about the temperatures, and if it is temperature is too much error, that also affect the coastal patches. That I checked when I did the experimental setup.
Thank you.
May I answer that question?
Yeah, sure.
We did this so far. Well, I think, yeah, trying to ask a very good question. What's the most important variables affecting this model simulation result, right?
Yeah.
If I answer this ocean circulation, it's a new one.
Yeah.
If you cannot get the ocean circulation correct, that offshore surf cannot be eliminated. This event is extreme event, so strong, the signal is so strong. You cannot lost if you initially put some sail offshore, they will be able to the shore if you have correct current. So this model right now, there will be a long way to go. Right now it's very crude. We assume that the nutrient levels, most of the background levels, no so offering specifically from people origin. So that nutrient is not considered at all. And also the biological process, very crude in this model at this time. There will be a long way to go. But we selected these features very nicely. Why? Because the main process is already represented in the model. This is coastal ocean circulation. That's the basis for this model, carbon model. We got to have correct ocean circulation model first. That's the message from this presentation.
So every physical geographer says that.
True.
All right, everyone, thank you so much. That was the last of our presenters. Please, wait, everybody, before you start talking, it's just some things to run by you. Come get your raffle tickets now if you want to participate in the raffle, because we're going to be doing it right after this. Please head to the MSL first floor lounge for the poster session at 3. There will also be a booth for the CMS science communication team. So please stop by for some new stickers and to see how they can help promote your research. And I think, what? Oh, Ryan, what am I? Yes, afterwards there'll be a TGIF. The poster session will go to 4:30 and there will be a TGIF in the MSL lounge for that. So please come join. And thank y'all for coming. Yeah, so thank y'all for